You know, I don't, I mean, the whole block fits into a particular amount of time. The division into individual lectures is based on content, not timing so much. So I don't actually know for sure.
All right, well, he'll come
Hour-ish.
He'll probably come an hour-ish.
I mean, if I need to change lectures, what should I do? Just hit escape. Right. Right. Move it up, pull it up, and then to start it new, it's just the.
It's just the recording that you have to
Change lecture to capture recording.
stop it and redo it for the next section.
All right. Well, we'll see.
He'll come in about a little bit before the hour.
Okay.
And wait for you.
If I have to do it before that, I'll punt. All right. Thanks.
If anything, just go through the whole thing.
It'll be okay.
Well, I don't have to.
I've done it before, but this is the new. It's a very spiffy new system. My goodness.
So recording's on and
All right, we're good to go. Start. Sorry. I'm not used to using a microphone. I'm used to projecting to the back of large classrooms. So, my name is Peter Hickmott, and I'm in the Department of Psychology here at UCR. Some of you probably recognize me. I recognize some of you. And I'll be leading you through a set of lectures on electrophysiology. Since I happen to be a professional electrophysiologist, odd as that may seem, this actually makes sense in a strange sort of way. So, if you're trying to contact me, for the most part, you're out of luck. But you can sometimes find me lurking in my office over in the Psych building in 2111H. If you actually go there and try and find me, you probably won't, because I'm spending most of my time in the lab. But you can call me. That's my extension, 7308. Probably the best way to contact me, not surprisingly, is by email. If you're wondering, I don't always check this email address that diligently after hours, so if you email me on a Saturday morning, which I'm sure you all will, you probably won't hear back until Monday morning during working hours. Okay? But if you have a question, feel free to contact me, really. I mean, I will respond, and I will try and answer your question in a semi-intelligible manner. In terms, I'd just like to thank Dr. Balcock over at UCLA, who actually provided a lot of the PowerPoint content here, which I'm stealing. So never make your own stuff when you can borrow. This is going to be interesting. I hope this isn't on timer. So in terms of the lectures, a fair amount of material, particularly figures, I extracted from a basic neuroscience text, which some of you have probably used. It's the book by Barrett, Connors, and Paradiso. This is going to annoy me. And there. And so if you're looking for more detailed coverage on some of the basic aspects of electrophysiology, this and some other books I'll point to later are actually good resources. This is a good sort of standard basic, next time I'm bringing my own pointer, standard basic book for this kind of stuff, okay? Alrighty, so your first question is, probably, why should we be studying electrophysiology anyway? Why do we care about bioelectricity and how it's generated and things like that? Well, the short answer is, of course, since I'm a neuroscientist, changes in membrane potential is pretty much how your nervous system does everything. This is where the magic is, folks. So pretty much everything you do, whether it's volitional movement, unconscious stuff, thoughts, feelings, the works, all runs on electricity. And chemicals, which we'll talk about also. But the dominant signaling mode is electrical in nature, as you know. Okay? And of course, it's also important to remember that changes in membrane potential are also important outside the nervous system for running all kinds of different things. So it's really important to understand this stuff at a very basic level, okay? Because you guys are going on to become doctors of various sorts, and this stuff runs an awful lot of stuff, okay? Now, in terms of the nervous system, unless you happen to be a single-celled organism, pretty much, you're going to have a nervous system, okay? And you need that nervous system to do things. So again, it's an extremely important basic process that certainly all vertebrates and hopefully all of your patients are going to use pretty often. They're all going to be vertebrates, I think. Maybe. In terms of thinking about clinical aspects of this, okay, this is very bad. I hate using the mouse. Hate. Sorry. Good suggestion, though. In terms of thinking of this clinically, this is not a neurology segment, don't forget. So we're not actually going to be talking about things like diagnostic issues, brain damage issues, or anything like that. You guys will get that later from actual clinicians who know something about it. But we will talk a little bit about certain aspects of pathology based on disruptions of normal electrophysiology, and there are quite a number. So again, in thinking about it clinically, it's important to have as knowledge in your toolkit. Also, when thinking about diagnosis, there are quite a number of diagnostic aspects that use various measures of electrophysiological processes, EKGs, EEGs, MRI, things like that. So we'll talk a tiny bit about some of those diagnostic issues at the end. Again, this is not explicitly a sort of neurology-based segment. It's really about basics. And then there are certain aspects of treatment that also will draw on understanding and better understanding of electrophysiology. So it really is important for you guys to understand this stuff. And I know that in many cases, a lot of what we talk about here is going to seem rather abstract and not particularly applicable to what you're doing. And that's true. But you're required to know it. And I'm required to teach it to you. And it is really important, right? Remember, speaking as a neuroscientist, all those other useful physiological systems, they're all there to keep the brain alive. That's all they're for, right? Right, the brain is the most important body physiological system. All right. I apologize to all of those who want to be cardiologists. But remember, the heart also runs on electrical potential. So it's really important to understand this stuff. Okay? By the way, feel free to break in with questions at any time. Just raise your little hand up, and I'll point in your direction. I know it's Monday morning, and you're all probably still zombies, but, you know, you could try. Okay. I'll come back and talk about this in more detail. I'll show this slide again. But I just want to sort of preface it, again, in thinking about this in a clinical context. How can we use electrophysiology in patients? And again, I don't want to talk about this a lot now. I really just want to show this to you as sort of an exemplar, a very quick exemplar of various sorts of recording methodologies that you might know of, and we will talk about some in terms of thinking about why electrophysiology is useful. So that includes extracellular type voltage recordings, recordings with surface and penetrating electrodes, things like EEGs, event-related potentials like ERPs, various functional magnetic resonance imaging methodologies, fMRI, optical imaging, which is really more research than clinical. And then on the sort of treatment front, again, a treatment avenue that's becoming much more important, particularly in a lot of psychiatric disorders, are various forms of stimulation of brain tissue, particularly things like transcranial magnetic stimulation, transcranial electrical stimulation, and various aspects of deep brain stimulation using implanted stimulating electrodes. And so again, if you're going to draw on these various diagnostic and treatment methodologies, again, it's really important to understand what is really going on. Okay? What are you really recording? What are you really manipulating? Okay? And that's really what we're going to talk about, very basic aspects of electrophysiology, membrane potential, action potential, et cetera. Okay? Okie dokie. All right, so again, in thinking about electrical signaling is used in very fundamental ways in really allowing for pretty much any sort of behavior. What I'm showing here is just a very, very simple sort of behavior. I am going to get this to work, really. Oh well, maybe I'll just point. So again, the idea is that when we think about even a very simple behavior, and what we're showing here is a very simple reflex arc, whereby if you have a painful stimulus, let's say to cutaneous receptors in your foot, your toe, so you stepped on a tack, for example, it is a tack, you will have a rapid withdrawal of the offending foot, okay? And so again, pretty much at every stage of this reflex, you're using electrical stimulation, electrical signaling of some sort. So at the very start of it, of course, you'll be using activation of cutaneous receptors. That's based on changes in membrane potential of the receptors. You're using things like action potentials to transmit the information along nerves, sensory nerves to the spinal cord. Then, of course, the action potential is converted to chemical signaling at synapses. We'll talk quite a bit about synapses as well. So that transmitter release activates both excitatory and inhibitory subpopulations within the spinal cord that basically cause certain muscle groups to flex, flexor muscles in this case, and of course, the antagonistic muscle groups are simultaneously inhibited. And so the contraction itself is also based explicitly on changes in membrane potential. So even in thinking about a very simple behavior, this simple reflex arc, we can see the very many important places where electrical signaling is used. Okay? And of course, if you're thinking about a more complex behavior involving hundreds of thousands or millions of neurons, then you just have to multiply that by several orders of magnitude. But at basis, it's the same stuff. Okay? Action potentials are fairly highly conserved, for example, over various sorts of neurons, and so on. So we're going to talk about these various parts, including action potentials, synaptic potentials, things like that, as we go forward. So that's really where we're going. And we'll come back and refer to this slide occasionally. We're not going to talk much about behavior, though, sorry. Okay? Okay. Oh, yeah, that too. So this is what we're actually going to talk about. The class is nominally boiled down into, at least conceptually, 6 or 7 lectures, depending on how you split it up. But basically, at the start today, we'll talk about basic electrical signaling, how it arises based on, excuse me, differences in the concentrations of various ions across membranes. We'll talk about some of the molecular channels involved. Then we'll talk quite a bit about action potentials. In terms of how they're generated and why they're useful. We'll talk a bit about synaptic transmission, again, which is really more of the chemical aspects of nervous system function. And then, as I said, right at the very end, we'll come back and talk a little bit about application of these processes. Tiny bit. All right. So this is stuff you already know. And to be fair, a lot of this is probably going to be review for many of you. But that's good. Review is always better than not. And basically, what we're talking about primarily in this set of lectures are neurons. So neurons are highly specialized cells of your body that make up the central and peripheral nervous systems, and they come in a hugely diverse set of forms and functions. I'm not even going to try and summarize different ways you might categorize different types of neurons into various subgroups based on structure, based on function, based on neurotransmitter composition. Suffice it to say that neurons are highly specialized cells, and they're primarily specialized for receiving and transmitting electrical signals. So a neuron is a very useful device for integrating information that impinges on its, say, dendrites and cell body primarily, and then transmitting that information via action potentials along its axon and communicating that information mostly by chemical synapses to various sorts of targets. So this is a fairly accurate drawing of a neuron. This is actually a more highly schematized drawing. It's actually a motor neuron. You can tell because it's interacting with a muscle, which is kind of an odd neuron. But again, showing really the main parts of the neuron, okay? And thinking about its structure. And again, like all cells, it's highly specialized. When thinking about a cell, this is always a good question I like to ask students. When you think about a neuron, for example, and it's different parts, it's fairly stereotyped parts, what part of the cell is the most important? I always like to ask this. It's an interesting question. What do you think? I need input. The soma is pretty vague. Anything like part of the soma? Considering how much stuff is in the soma. Nucleus. I hear some nucleus. Any other thoughts?
Plasma membrane.
Plasma membrane. See, he's got the answer I'm looking for. When you think about it, and nucleus is a pretty good answer because, of course, nucleus is where the genetic material is, which is very important. However, my take on that is the plasma membrane is more important because without a plasma membrane, you ain't got a cell in the first place, right? You need something at a very fundamental level to separate in from out. It allows you to manipulate your intracellular environment in many very important specialized ways, okay? So in order to have cell biology at all, you got to have a cell. Okay? Any cells in your body that don't have nuclei? Name one.
Red blood cells.
Red blood cells. Name another. This is a tough one. Platelets, kind of. Name one other one that I bet you won't think of. No? And you've all heard of the endobiome, right? All those symbiotic bacteria type things you got in you, very important, very current these days. A lot of those are prokaryotes. Prokaryotes don't have nuclei. Ah, I didn't say they were necessarily your cells, did I? But do not underestimate the importance of those endosymbionts. Okay, we're not going to talk about them at all, but it's still very important. And I find that to be a very interesting question for you guys to think about. Don't forget that. Anyway, all right, so moving back to neurons. So in general, and again, this is a generality that is very variable. Generally, neurons receive inputs on their dendrites and stomata. So these are dendrites, of course, which are projections from the soma. Soma itself is the cell body, where the nucleus is, where protein synthetic machinery is, and all that stuff that you already know. In general, they integrate that information at the spike initiation zone, also called the axon hillock, which is a point typically right at the transition between the cell body and the axon. And so it's really a lot of what's going on in terms of output has to do what's going on in the spike initiation zone. Okay, and we'll talk some more about integration when we talk about synaptic potentials. Output the information via action potentials. Action potentials are the primary sort of information carrying potential that neurons use to send information over long distances. And we'll see why the properties of the action potential allow that to happen. And then, of course, influence their targets, primarily via chemical synapses. Although there are also so-called electrical synapses, gap junctions, which actually act pretty much directly on targets. So we'll talk a little bit about those. Okay? If you can't show your own work, why bother? This is an actual neuron from the rat cerebral cortex, actually primary somatosensory cortex, filled with a molecule called biocytin, reacted for a fluorescent chemical reaction. I filled the cell myself after recording from it. And it's just a nice example of the complexity of a fairly typical type of cell that you would find very similar sorts in your own cortex. So again, you can see this is pretty much everything you can see is pretty much all dendrite. The axon's too small to be seen at this resolution, pretty much. You can see it a little bit. And it's just a good sort of demonstration of the complexity of neurons. Okay? They come in, as I say, in a huge variety of forms that drastically affect their functions. So, and we'll talk some about that. Okay? Besides, it's a beautiful picture, right? Right. Before we go diving into the world of neurons, which is what we will talk almost exclusively about, I do not want you to forget about the other sort of main non-vascular cell within the nervous system. Vascular is also very important. We won't talk about that pretty much at all. That, of course, are glial cells. And if you were a disinterested observer and you looked at a human brain, or any kind of vertebrate brain, and you just counted the number of cells, you'd actually find there are a lot more glial cells than neuronal cells. You'd probably assume that glia are actually more important than neurons. There's probably 1 to 10 trillion glial cells in the nervous system, as opposed to maybe 100 billion, that's for a human nervous system. And so the study of glial cells is in many ways still in its infancy. Okay? There's an awful lot we don't know about glial function, but the more we find out, the more important it seems to be for understanding normal neuronal circuit function. So we don't have time to talk a lot about aspects of glial physiology and glial impact on nervous system function, but I don't want you to forget about them. And don't forget, just as neurons have multiple forms and functions, the same is true of glia. Glia are divided up into sort of 4 major categories. Astrocytes, which you'll find primarily in the central nervous system, oligodendrocytes and Schwann cells, both of which are responsible for myelin. Oligodendrocytes in the central nervous system, Schwann cells in the peripheral nervous system. And then there's microglia. Microglia are very interesting glia. They've been mostly characterized in the context of neuroimmune function. They're the major immune cells of the central nervous system in the brain. As I said, it's unfortunate that we don't have time to talk more about glial cell functions because, as I say, the more they're studied, the more they seem to be important for normal ongoing nervous system function. And their functions are also clearly perturbed in certain pathophysiological states, including things like epilepsy. They may even be causative for some of those. Again, there's a lot of interest in that. But we're really going to focus in on neurons. Yes?
Sorry, what did you say astrocytes do?
I didn't. They do a great many things. Historically, astrocytes have mainly been associated with what you would think of as housekeeping functions. That is, particularly the uptake of various sorts of chemicals, including neurotransmitters, which we will talk about. It's clear they also release a large number of molecules that are biologically active, including neurotransmitters. So they have a wide variety of functions, honestly. So we're not really going to talk about all their possibilities. So that's my answer. As I said, we don't have time to talk about all of them, but we will come and talk a little bit about neurotransmitter reuptake, and that is mainly the province of glia, and in the central nervous system, that's mainly astrocytes. So, other questions? Yeah, I'm going to go quickly, as you may have noticed. So it's important if you miss something, if you have a question, to raise your hand up. We have a lot to cover. All right, so to understand electrical signaling, which we're hoping to do, first we really have to understand the components that give rise to the electrical differences across the membrane. And that's really what we're mostly talking about here, is transmembrane potential. What's going on right at this membrane? So again, really asserting the importance of the plasma membrane, because that's really what this is mostly about. What's going on right at the membrane? Pretty much everything we talk about in terms of changes in membrane potential, it's really localized to the membrane. Okay, if you go to the sort of cell body, that's pretty much electrically neutral. Anyway, so we're going to talk about chemicals that are involved, both in the cytosol and extracellular fluid. We'll talk a little bit about the lipid bilayer, although I'm assuming you already have covered quite a bit of that, but some of the more important components of that, and the proteins embedded in it that are going to be important, particularly, of course, ion channels of various sorts. We'll talk a tiny bit about ion movement, ion diffusion, and how that generates electrical potentials. Again, this is not a chemistry class, so we will not go deeply into that. You certainly could. And then we'll talk most importantly about the ionic basis of the resting membrane potential. So the ideas of equilibrium potentials, which are determined by the Nernst equation, which we'll define, ion distributions across the membrane, and ion permeabilities. And again, the focus here is at rest. We'll talk about active processes like action potentials in the next couple of lectures. Okay? All right, so cell membranes, again, very important. Really, if you don't have membranes, you don't have life as we know it. Maybe you could have something else, I don't know. They appeared, again, somewhere 3 billion plus. As they find better fossils, they keep pushing the date earlier and earlier, so who knows? It's probably 4 billion years now. And again, it allows you to separate in from out, so you can actually have an intracellular environment. So you can do something different, really, from what's going on in your environment. So, again, if this is a typical cell, it's got an inside, it's got an outside, it's partitioned by this plasma membrane. And in thinking about what is most important, if you will, in these environments, well, probably the most important chemical is water. Pretty much, by far, the vast majority of intra and extracellular environment is just water. 70% by weight of a cell, just about. Water, as you know, is a really remarkable substance. I'm not going to go into water chemistry, thank goodness, but again, the basic idea, one of the things that makes water so useful is that to a certain extent, water is a very important solvent. Okay? It dissolves a lot of different things. It's polar. So, of course, it dissolves polar species. And what we're most interested in are ions. And ions we'll talk about are things like sodium, potassium, and chloride. Those are actually, we'll talk about these are the ones we'll talk about almost exclusively. Sodium, potassium, calcium, and chloride. We can ignore most of the other ones, thank goodness. So, talk a little bit about ions in solution. We'll talk quite a bit about that. So ions, of course, are atoms or molecules with net charge. And in terms of biological molecules, that means almost all of them, except some of the lipids. The net charge, of course, is the difference between the number of protons and the number of electrons. You've all covered this until you're blue in the face. For a monovalent ion, the difference is 1. For divalent ions, the difference is 2. So, of course, calcium is a 2+, divalent. Sodium is a monovalent plus. Chloride would be a monovalent minus. Cations are positive. Anions are negative. So, of course, when we talk about electrodes, and you talk about batteries, anion and the anode and cathode are reversed, but oh well. In terms of our interests, we're interested in a very small variety of these ions. Sodium, potassium, calcium, chloride. And again, remember that in a typical solution, these ions, of course, exist as hydrated molecules. Okay, they're surrounded by a shell of water molecules, as is shown here. we're not going to talk a lot about that, about the hydration state of these ions. But it actually is important for how they pass through the entities that we're going to call ion channels. And again, we're not going to go very deeply into the physical chemistry of it, but suffice to say that the hydration state, and actually in many cases the thinking is the hydration state is actually changed during the passage through an ion channel. So that is actually important for those of you who are physically inclined. Okay, hopefully this is all review. That's why we're going quickly. All right, so for a typical neuron, which might look something like this, inside, outside, maybe, what are the concentrations of the ions that we're talking about here? And so fortunately, we have a very limited number of ions, and it's only important really to keep track of what's going on in and out. And you could memorize these concentrations if you wish, but what's really important to think about is the sort of the relative amounts. This ratio is actually quite useful. In terms of potassium, the basic idea is that it's high inside, low outside. For sodium, it's the opposite. It's high outside, low inside. For calcium, again, it's pretty low on the outside, but it's incredibly low on the inside. Cells actually keep track of their calcium concentrations very, very carefully. There's a very highly regulated molecule. That's because, in terms of functions inside cells, calcium does a million things. It's a very important second messenger molecule, acts on all kinds of important intracellular cascades. And of course, if it gets too high in a cell, it will kill the cell in many cases. Chloride is mostly highly concentrated outside and not so concentrated inside. And again, the key points here, again, are these, really. More K inside, more sodium, potassium outside. For chloride, the issue for chloride is a little bit more complicated. And again, I don't want to get into a lot of detail. There's not much chloride inside, actually, and you'd expect more to balance out the amount of, say, potassium. But in order to balance out the charge, cells contain a lot of anions of other sorts. Bicarbonate, phosphate, most of your proteins turn out to be net negative charge, nucleic acids. And so there are a whole variety of great big molecules inside cells that ultimately maintain overall electroneutrality. We won't talk a lot about them, but for those of you who are wondering, that's how it's done. In a large volume, say within a cell or outside the cell, the number of ions, cations and anions is about equal, okay, electroneutrality. At the local scale, that does not have to be true. And again, that's really what we're going to be focusing in on, this very small microenvironment by the membrane, where we will get moderately sized potential differences, okay? And we'll talk a little bit later about how do these differences arise? How do cells maintain these so-called concentration gradients or concentration differences? Okay, which you probably already covered. You've already covered pumps, basically, right? Membrane pumps? Yeah, so you already know the answer to that. So you already know this. You've probably actually seen some slide very close to this. In terms of the lipid bilayer, which you know, it's a phospholipid. So it's a bilayer of phosphate groups on the outside with lipid tails on the inside. And probably the most important thing to remember about that, of course, is it's fat. It's a lipid. Polar molecules do not pass through it very readily. And for our purposes, the more important polar molecules are ions. Ions do not pass through the plasma membrane without some kind of help. Okay. That's not true for other sorts of molecules, particularly, of course, hydrophobic molecules, oxygen and CO2 being very important ones of those. Some of the small uncharged polar molecules can pass through it moderately. But again, for our purposes, for ions, we're going to say it's pretty much impermeable unless there are specific channels or processes that allow the ions to be carried through the membrane. And again, those may be passive processes, particularly ion channels like these here. In some cases, they may be carrier-mediated. And in some cases, they may actually be subject to active pumping using ATP as energy. Okay. So again, that's going to turn out to be very important for setting up these charge gradients that we're talking about. And those charge gradients are going to be what set up the biological potentials we're interested in. Okay? All right, you already know this stuff. But just to remind you, ion channels, which we will talk a lot about, are relatively ion selective. By relatively, that may be very selective for a single ion like sodium, or promiscuously selective for something like cations, like sodium and potassium. We'll see both of these sorts of things. Channels can have different states. Some channels are pretty much always open, so-called leak channels, for example. Some of them can fluctuate between various types of open and closed states. And when they're closed, of course, they don't allow ions to pass through them very efficiently. When they're open, they will allow their relatively selective ions to pass through. In terms of the chemical structure, I'm not going to go there. Okay. What is important to remember is that for channels with gates, those gates have very specific stimuli that will allow them to open or in some cases make them close. That is going to be very important for understanding, again, ion flux through the membrane, particularly voltage-gated channels and ligand-gated channels. Okay, I'm not going to talk about structure. I don't care about structure. Right? Right. Ion channels are diverse. You know this also. But in terms of thinking about the class and what we're going to talk about, we're going to talk quite a lot, especially at the beginning, about voltage-gated channels, which again are typically opened by depolarization of the membrane, which is a term we'll define later. You probably already know it. And later on, when we talk about synapses, we'll talk quite a lot about so-called ligand-gated, chemically gated channels, particularly ones that are gated by chemicals in the extracellular space. So again, these are channels that open in the presence of particular molecules, like neurotransmitters. We won't talk about these subtypes pretty much at all. We won't talk very much about channels that are gated by intracellular chemicals, and we won't talk very much about mechanically gated channels, although those, of course, are very important for things like mechanical transduction events, say, which we talked about at the level of the sensory receptors, just for example. Okay? All right, so we talked mostly, very generally about our chemicals: cytosol, extracellular fluid, water, ions. Phospholipid bilayer, impermeable to ions, except where there are ion channels. Those are really the main points to come away from there. And I suspect you already knew that. So let's talk about ion movement, diffusion, and electrical potentials. And then we'll talk about the ionic basis of the resting potential. Diffusion basics. So diffusion is a very fundamental chemical property. Anything that's dissolved in anything else will be subject to simple diffusion. Ions are no different. So ions dissolved in a solution, say sodium and chloride, when you take your typical table salt and drop it in a cup of water, you get salty water. The saltiness, of course, is based on sodium and chloride ions dissolved in solution, mostly sodium. Although a lot of table salt, of course, has iodine in it, and a lot of it has potassium too. Well, we will talk about that. So in terms of diffusion, the main thing to remember is that ions in this case, or any chemical that's dissolved, will tend to go from where it's at a high concentration to a low concentration. Okay? It's just a stochastic, probabilistic process. This will happen with variable kinetics, depending on things like temperature, size of the molecules, permeability of the say, the membrane or the channels. But the basic idea is eventually, if you have, say you have sodium chloride on one side of an impermeable membrane, hello, I'm just going to point, excuse me, and you have this, say this is a lipid bilayer right here. The lipid bilayer, of course, is impermeable to ions, and so this is a stable situation. You're going to have sodium chloride here and water here. If you insert channels into the bilayer, so a sodium permeable one, a chloride permeable one, then you're going to get diffusion. Okay? The ions are going to diffuse down their concentration gradient from the high concentrations to the low concentrations, like this. Bring in my own pointer next time. So you'll have diffusion. Okay? Diffusion will continue until they are basically equally distributed. We'll see why that isn't entirely true in a second. And that breaks down the concentration gradient. The concentrations will be the same on either side of this membrane. Okay? And that's basically what's going to be important. So concentration gradients are going to turn out to be really important. Okay? Concentration gradients are going to drive the generation of membrane potential, and they're actually going to allow us to change membrane potential by changing the relative permeability of the membrane, as you probably know. Okay? Okay. Now, if we were just talking about uncharged molecules, of any sort, we wouldn't have to worry about electricity. But since we're talking primarily about ions moving through channels in solution, we have to worry about electrical potentials, because of course ions are charged. Anytime you got charges in motion, you're generating current. And that's, as I say, going to be really important for the generation of the membrane potential. So basically everything is made up ultimately of charged particles of some sort or another. I guess if you dice them small enough into quarks, they're not technically charged, but let's just leave it at that. Yeah, I know, we're all made up of probability functions of quarks. Okay, well, you can't study much at that level unless you're a physicist. So we'll talk about ions as if they're just charged particles. Most things are electroneutral because they contain basically equal amounts of charge. That's the easiest way to think about it. 1 mole of sodium chloride, chemistry coming back in chunks, contains Avogadro's number, N, of sodium ions and an equal number of chloride ions. And everybody remembers the value of Avogadro's number? Of course you do. 6.02 times 10 to the 23rd particles. Yeah, I remember that. You are much closer to basic chemistry than you probably do. You probably memorized it for your MCATs. Good job. Now you can forget it, because you're never going to use it again, probably. I don't. Quantity of charge is measured in coulombs, C. The elementary charge, which is the charge of a proton, is another number I'm sure you remember, 1.6 times 10 to the minus 19th coulombs. Okay, so what? The charge on Avogadro's number of elementary charges is called the Faraday constant, F. So it's 6.02 times 10 to the 23rd times 1.6 times 10 to the minus 19th, which is ballpark 10 to the 5th coulombs per mole. That is the charge on 1 mole of sodium ions. Why am I telling you this? I don't know, but I'm telling you this because these values will actually appear in the equations we're going to talk about that define things like the Nernst equation. Okay? I actually don't care if you know the actual numbers involved. If you want to memorize them, feel free. But as I say, actually, I think I might have been able to come up with Avogadro's number, but that's about it. So the charge, of course, on 1 mole of chloride ions is negative F, Faraday constant. And of course, if you're divalent, you have 2F, or 3F if you're trivalent, for example. As I say, the main importance for this is that these various terms like N and F and C are going to appear in some of the equations that I am going to want you to know. And it's important that you have at least an idea of what they are. Okay? That's the reason I'm running through this, really. Okay? Just definitions. All right, more basics. Physics and chemistry. All right. Again, we're talking about electricity in very basic ways. We're not going to talk about it in a huge amount of detail. But it's important to remember, of course, that since ions are charged, they will, of course, move in an electric field. And, of course, as you remember, opposite charges attract, like charges repel. Plus repels plus, minus attracts plus. So there will be net movement of sodium to the cathode, chloride to the anode. So remember, anions, which are negative, move to the anode. Yes, I know. And cations move to the cathode. That's the way to remember it. You can remember anions are negative ions, anion. That's how I learned it back when I was a wee young lad. In terms of thinking about charge, again, what's really important for our purposes is charge in motion, ions. That, of course, is current. So any kind of charge in motion, whether it's electrons in a wire, which you have all over the place, or in our case, ions through ion channels, generates an actual current. Okay? In terms of definition, all this is arbitrary, but so current is defined as positive in the direction of positive charge movement. So in this particular example, where we have sodium ions moving in this direction, towards the cathode, chloride ions moving towards the anode, the positive current flow is the direction of sodium, positive ions, towards the cathode. Oh, man. It's going to drive me insane. You come up here and do this and see how you do. All right, never mind. Anyway, the positive current flow in the direction of sodium from anode to cathode. So when we're talking about current as positive, we're talking about positive charge movement. Okay? Of course, then if your charge carrier is negative, it's going to be the opposite. The amount of current flow will depend on the potential difference and the electrical conductance. So the potential of this battery, how big is it, and the ability of, in this case, ions to move within whatever solution they are, the conductance. Also, of course, which is also synonymous with the inverse of resistance. Potential difference is measured in volts, of course, as you know, and is the work needed to move a unit test charge from one point to another. Don't you know? Okay. To move 1 coulomb of charge across a 1 volt potential difference, need 1 joule of work. That's, again, just the definition. The main sort of intuitive point, which is what I care more about, more current flows as voltage is increased. So as you increase the potential difference across here, you're going to have more current, all other things being equal. The ease of current flow between 2 points is called conductance, small g. In this example, conductance can be increased by adding more salt. So it turns out that the higher the concentration of dissolved ions in a solution, excuse me, the higher its conductance. Pure water is actually a fairly poor conductor. If you have distilled water with no ions in it, nothing dissolved in it, it's actually not a very good conductor. But anytime you get salt into it, it becomes quite a good conductor. Okay? That's why you don't want to swim when there's a thunderstorm. Okay? Really. People in California don't seem to appreciate that. If a lightning strikes the sea or the lake you're in, it will be conducted to you very efficiently by water. Anyway, so in this case, conductance can be increased by adding more salt, more ions, by using ions that are more mobile, which of course we can't actually change necessarily, or by moving the electrodes closer, so distance will also matter. Conductance is measured in Siemens, S. Resistance is measured in ohms. Of course, resistance is just 1 over conductance. Okay, there are very few formulas that I'm going to want you to know. This is one of them. I'm assuming you probably already know it, Ohm's law. Ohm's law is the relationship between current, voltage, and conductance. V equals IR. Voltage equals current times resistance, or current equals conductance times voltage, excuse me, which is the same. So if the conductance is 0, no current can flow, obviously, even if there is a very large potential difference. If the potential difference is 0, no current will flow even though the conductance is high. Okay, and we'll see why that's important as we go forward and talk about occurrences at the membrane potential. So a situation more like this. Because it turns out, of course, that the membrane conductance in many cases is regulatable from relatively impermeable to relatively permeable. And that, of course, is then going to affect current flow, which is going to affect voltage. Okay? Okay. You can memorize Ohm's law. That's a good one. And also the derivation of Ohm's law that contains driving force, which we'll talk about in a second. All right, so we've talked a little bit about basics of ion movement, diffusion, and electricity at the level that we're going to go over it on. If you want more, feel free to consult a physics textbook. Let's talk about the resting potential. That's really why we've talked about all this stuff. What happens at membranes? How do we put together conductance, ion concentration gradients, electrical gradients, to decide why is it that neurons have the resting membrane potential that they do, and later on, how do we manipulate it? Okay? Okay. If you look at a typical cell, and you want to record the resting membrane potential, when we're talking about the resting membrane potential, we're talking about a potential across the membrane. So in order to record it, you have to have a sensor, an electrode, inside the cell, recording with respect to a ground wire outside the cell. So you got to stick a very fine electrode through the cell membrane into the neuron, which has maybe a 10 or 20 micron cell diameter. So it's, technically it seems like it should be very difficult, but actually it's not. I do it every day, or most days. And so when you do that, you will record a membrane potential. This is not just true of neurons. Pretty much every cell you've got has some kind of membrane potential. And different cells actually may use that for different things. For neurons, we're going to use it for information. That's the important thing. The membrane potential is the voltage across the plasma membrane. The resting membrane potential is the voltage across the plasma membrane at rest, in the absence of other influences. It's usually abbreviated as V sub m, Vm. In a typical neuron, the value of the resting potential can vary drastically. Different types of neurons have different resting potentials. An individual neuron can change its resting potential based on a whole variety of modulatory influences. But a good ballpark figure to keep in your mind is somewhere around minus 65, minus 60 millivolts, okay? That is a good ballpark. In actuality, it might range from anywhere from maybe minus 40 millivolts to minus 70 or 80 millivolts. And so that's a pretty good ballpark figure. And so what that means operationally is there's uneven distribution of charge across the membrane. Okay, right at the membrane, you look right here in this microenvironment, you'll see this separation of charges. Okay, negative inside, positive outside. Okay. Remember, the cell as a whole is electroneutral. It has pretty much the same number of ions, of anions and cations, but locally you can build up this relatively large membrane potential. Okay? Okay, where does it come from? And why is it the value that it is? Well, again, if we think of a very simple example where we have two compartments divided up by an impermeable membrane, like a plasma membrane with no channels, and we just have potassium and some kind of anion. Doesn't matter what kind. Could be chloride, could be proteins, could be some mix of them. And we have different concentrations of these molecules. So we have a high concentration of our Ka inside this cell and a low concentration outside, which is what we see in normal circumstances at neurons. All right, so what we've got is a very large concentration gradient, just sitting right here doing nothing. Why is it doing nothing? Because there's no permeability. It can't do anything. Okay? The system is stable. There can't be diffusion because there's no permeability. The hydrophilic ions will not move. So the potential difference, of course, is zero. Okay? There's no current. The conductance, if you will, is basically zero. All right, well, that's boring, unless you like salty water. So let's make it a little bit more interesting and a little bit more accurate as to a neuron. Let's put a potassium selective channel inserted into this bilayer. So potassium is going to be able to diffuse down its concentration gradient. All right, well, what that means is, of course, when you first insert that channel, potassium ions are going to move down their concentration gradients from this side to this side, from in to out, as we're calling it. The anion cannot move because, of course, the membrane is impermeable. Okay, so its concentration does not change. It can't cross the membrane. All right, so what? Well, remember, if these were uncharged molecules, we wouldn't have to consider charge. Okay, that was a circular statement. We wouldn't have to worry about potential. We wouldn't generate a potential. But since these are charged molecules in motion, again, we're generating current. But the other thing to remember is every time we move a potassium ion down its concentration gradient from in to out, the inside is going to become a little bit more negative with respect to the outside, or the outside becomes more positive with respect to the inside. So a net negative electrical potential is generated on the inside because those anions are left behind. Okay, well, don't forget that by simple electrostatics, as we're building up negative potential here, that's going to tend to attract potassium ions towards the inside of the cell. So we really have two different forces acting on a potassium ion, say right here. One force is the concentration force, which is driving it in this direction, from in to out. But as we build up, as potassium ions move more and more, you generate a larger and larger force attracting it in the opposite direction, from out to in, based just on the electrical attraction of potassium ions for this negative potential inside the cell. Okay. At a certain point, those two forces are going to be balanced. The net force driving it out via the concentration force is going to be equivalent to the force, if you will, driving it in based on the electrical gradient or the electrical potential. What that means is there's a point where the outflow of potassium down the concentration gradient is equal to the inflow down the electrical gradient. This is the so-called equilibrium potential, okay, where there's no net potassium flow. Okay. That's going to turn out to be the point at which the resting potential is generated. That is why you have a negative potential inside. And that is why it is about minus 65 millivolts. Okay. And we'll see how that value, how we can arrive at that value of minus 65 in a couple of minutes. Okay? But this basically is how it's done. It's all about balance of forces in terms of the concentration gradient and the electrical gradient. Okay? And in this situation, it's a stable system. Okay? It will reach equilibrium. It will stay at this equilibrium unless perturbed, basically forever. So this is how, and this is basically how membrane potential is generated. Resting potential is generated. Because as we'll see at rest, this is approximately the situation that we have, because at rest the membrane is quite permeable to potassium, but not terribly permeable to things like sodium or calcium, okay? Which we'll talk about in a sec. Does everybody get this? This is the magic right here. Ion chemistry, very important. There you go. All right, some key points. So-called leak potassium channels give rise to this negative membrane potential. So in a neuron, normal cell, you have a bunch of channels that are open at rest, potassium channels. Those are the ones that allow for this to happen. Only very small amounts of ion flow are needed to create potential differences, okay? Again, it's a very localized phenomenon at the plasma membrane. And you can calculate it using various methods, which I don't want to go into. But given a typical, say, 50 micron diameter cell, if it's this is assuming a spherical cell, which is not true. But if you have 100 millimolar calcium outside, potassium to generate the potential difference of minus 80 millivolts, again, the actual values don't matter much, but the point is that to generate that potential, you only need a concentration change of 0.00001 millimolar. So that's what? A very, very tiny fraction of a percentage of all the available potassium ions. Okay. That's important to remember. These changes in membrane potential are generated by a very small fraction of the available ions. Okay. That's the point from that. So net difference in electrical charge occurs at the inside and outside surfaces of the membrane. Therefore, the membrane stores electrical charge. Okay. A property called capacitance, which we will talk about some more later in terms of the membrane. Ions are driven across the membrane at a rate proportional to the difference between the membrane potential and the equilibrium potential. Okay, this is an important function right here, ionic driving force. Okay. Turns out we're going to substitute that driving force term into the Ohm's law equation to allow us to understand ion movements when we have, again, this E ion, this so-called equilibrium potential. That's going to be really important to understand because that's going to allow us to understand how ions are going to move in various situations. Okay? And we'll talk more about that particular equation in a second. If the concentration difference for an ion is known across the membrane, then you can calculate the equilibrium potential using the Nernst equation. So the value of that equilibrium potential, which we just talked about here, is determined strictly based on the relative concentrations of permeable ions. Okay? In this case, just potassium. All right, so let's just think about this for a second in the context of Ohm's law, and to think a little bit about current movement. And we'll come back and talk about the Nernst equation in a sec. All right, by convention, just because people wanted to do this, the direction of current refers to the direction in which positive charges are moving. I'm going to try this again. Okay, so in terms of a cell, when we talk about inward current, we're talking about positive charge moving in from the outside. So if you have, say, potassium ions moving from the outside of the cell to the inside of the cell, that is inward current. However, you can also generate an inward current if you have negative charge carriers moving from the in to the out. Don't forget, everything is relative. So, and when we talk about representing current, when we talk about physiology traces, we're going to talk about inward current as downward deflections, outward current as upward deflections. Again, that's just conventional. That's just the way that, you know, physicists and physiologists decided it would be. Doesn't have to be that way. So if this is current, inward, outward. The issue is, just to allow you, let you know some of the complications, when we talk about voltage across the membrane, an inward current like this is going to generate a positive change in voltage, right? Because positive charges are moving into the cell, correct? We're going to represent a positive voltage change with a positive deflection on our oscilloscope. Yay. So if you have an inward current, which we're going to represent as this downward deflection on our oscilloscope, that's going to correspond to an upward deflection in voltage, which is very confusing. This, of course, will correspond to a downward deflection voltage. I tell you this now because we will be looking at quite a lot of traces, basically oscilloscope traces of things like membrane potential and membrane voltage, membrane current. It's important to remember which is which and what the direction is telling you. And I'll try and keep telling you that because it's kind of hard to remember. Anyway, inward current is shown as downward traces, outward current as upward traces. We'll see quite a lot of those things. All right, let's get back to Ohm's law, and let's think about directions of current flow. And let's think about how we're going to factor in this sort of this driving force idea. And actually, really the equation that's important, again, this is Ohm's law as we're talking about, current equals conductance times voltage. Well, when we're talking about this situation where we basically have a battery in series, that's what the equilibrium potential is, you got to factor in this equilibrium potential in terms of your voltage term. This is the so-called driving force on a particular ion. So the current for an ion is the conductance for that ion times the driving force. Okay? The driving force is the current membrane potential minus the equilibrium potential for that ion. So the thing to remember, well, we'll come back to that in a second. So for, say, potassium, which we've been talking a lot about, again, the current that we're looking at is going to be the conductance for potassium times the driving force. So let's think about this for a second. If we have a potassium equilibrium potential of minus 90 millivolts, which is actually pretty close to a typical value, okay? So if this value is minus 90, EK, if the cell is sitting at negative 10 millivolts, so it's a little positive from rest, which way is current going to flow? Which way is current going to flow? Yeah.
Into the cell.
Well, let's think about it. Let's just parse it out in detail. So you've got a membrane potential of negative 10. You've got an equilibrium potential of negative 90. Negative 10 minus 90 is plus 80. Assuming we have conductance. If we have no conductance, we have no current. Assuming we have some conductance, we have plus 80 for our, our driving force. So, all right, so we're going to tend to get that. Outward current, sorry. I got confused for a second, as I so frequently do. One very important point is here, if the equilibrium potential is minus 90 and the cell is sitting at minus 90, which way will current flow? The basic answer is correct, both ways equivalently. So there is actually no net current flow. Okay? For every potassium that goes inward, a potassium will go outward. So one of the easiest ways to think about the equilibrium potential is this is the potential that the membrane wants to be at for this particular ion species. If you are at that membrane potential, you have no net current. Okay? That's sort of clamped at that membrane potential. If your membrane potential diverges from your equilibrium potential, current is going to flow, and current is going to flow to bring you back to that equilibrium potential. Okay? So if you move the equilibrium potential to a positive potential, you're going to generate a current that brings it back down towards minus 90. Okay? And vice versa. If you move the equilibrium potential in a more negative direction, potassium will flow such that you generate, such that you generate a current that, again, brings you back to that equilibrium potential. So the equilibrium potential is sort of you can think of it as a clamp, if you will. And it's the potential that the membrane is trying to be at, or will try and generate current to sit at if there is some change in membrane potential. Okay? And at rest, of course, at the resting potential, this driving force is zero. That's the whole point. So there's no net current flow. Okay, is that clear intuitively? Because we're going to talk a lot about current flow in various directions. And the important thing is going to be able really to intuit which direction current flow is going to be in a particular membrane potential environment with particular ion permeabilities. Okay? Okay. Benner's equation. Okay, so I told you we'd find some of these values that we defined. RT, ZF, okay, R, of course, is something. The gas constant, a different constant. T, absolute temperature in degrees Kelvin. Z is the charge on the ion we talked about. F is the Faraday constant. Ln is the natural log. This is the concentration of the ion outside, concentration of the ion inside. That will give you the equilibrium potential for that ion. So as you can see, the equilibrium potential depends on things like temperature, of course. It depends on things like the valence of the ion we're talking about. But the main thing that it depends on is this. Whoops. the relative concentration of the ion outside versus inside. So what that means is basically that was really implied, if you will, or determined. That's going to tell you basically where the resting potential is. Okay? Because at rest, the membrane is pretty much only permeable to potassium. And so the resting potential it approximates the potassium equilibrium potential. As we'll see, it's not exactly equivalent to the potassium equilibrium potential. But we'll talk about that in a sec. So at equilibrium, the whole point is there's balance. There's balance between diffusion pushing ions down the concentration gradient and the electrical gradient, which causes the ions to move towards their opposite charges. So increasing thermal energy, temperature increases diffusion, increases the potential difference. For biological systems, for the most part, that doesn't matter. Well, for warm-blooded vertebrates in particular, because you keep your temperature pretty constant. If it varies too much, that would do bad things, as you know. And of course, increasing the charge will decrease the potential difference needed to balance diffusion, because again, it's going to affect the electrical gradient. Again, that's an intuitive analysis of it. Do I have a value for this? All right. Blah, 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 blah. At 37 degrees centigrade, this is a useful value to have. It's actually useful to convert this whole Nernst equation from natural log to log base 10. And it turns out when you do that, you end up with 2.303 RT over F as this constant. And we're going to assume this is a constant. Okay? We're going to assume that temperature is constant, which is a fair assumption. So at a typical approximate body temperature of 37 degrees C, RT over F times 2.303 is about 61.54 millivolts. Okay? So again, that constant is going to then be multiplied by the log of the K over Ki to get you the overall, the overall membrane potential. Okay, so let us recall our concentrations here. For potassium, which is what's most important for the resting potential, the out to in ratio, it's highly concentrated on the inside, low concentration on the outside. So out to in is about 1 to 20, 1 versus 20. And it turns out the equilibrium potential, again, for pure potassium is about minus 80 millivolts. Okay, these are useful values to have in your mind. Okay? I would say you should probably have at least a pretty good ballpark figure for these equilibrium potentials for these 4 ion species. Because that's really going to determine much of our ion movement, much of our current. Sodium, of course, is the opposite in a way in that it's highly concentrated outside. Its ratio is about 10 to 1. So its equilibrium potential is about 62 millivolts. Calcium, since its difference is so high, has a relatively high positive equilibrium potential. And then chloride has a kind of a mid-range negative equilibrium potential. And it turns out, as I say, in terms of the biological systems, these 4 ions are pretty much it. So when thinking about membrane potential, the potassium equilibrium potential is just about the negative limit normally on membrane potential. That's as negative as it's going to get. Calcium is about as positive as it possibly could get. Okay? So that's good to know. It's good to put a bracket, if you will, on the amount of membrane potential change we can have, somewhere around 200 millivolts maximum. As we'll see, we pretty much never get that much. But we will approach 100 millivolts and change. Okay? And again, depending on the relative permeabilities of the membrane to these various ions, that's going to determine the overall membrane potential, as we'll talk about when we talk about the action potential. Okay? At rest, again, potassium dominates. Permeability to potassium is very high. There is some permeability to sodium and some of the other ions. That is why the resting potential is not exactly the same as the potassium equilibrium potential. Okay, there's a sodium current involved in it in particular as well. Okay? So there's an important sort of implied question here. Why do cells have negative resting membrane potentials? I mean, what serves to set up those concentration gradients in the first place? I mean, we started out when we were talking about ions in biological systems with these concentrations. I told you potassium in, potassium out, sodium in, sodium out. Why are they the way they are? Why are they those concentrations? Because those concentrations are really important. Well, as you hopefully know, these ion concentration gradients are built by cells. And cells actually spend an awful lot of energy, particularly working that sodium-potassium gradient. So it turns out, in order to have this as a stable system, you have to use energy. And there's a pump, which I'm sure you've heard of, called the sodium-potassium ATPase, or the sodium-potassium pump. And basically what this pump does, it lives in the membrane, particularly of neurons at very high concentrations. And when it cleaves a molecule of ATP, excuse me, it transports 3 sodium ions from in to out and 2 potassium ions from out to in. That is how these concentration gradients are maintained. Okay? This is a highly energetically costly process, keeping these concentration gradients built. Somewhere around 2/3 of your neuron's energy is taken up by running this pump, keeping that membrane potential charged, basically. So, with that in mind, let's say you use a blocker of this sodium-potassium pump. There are several. One of them is called wabane. What will happen to the resting potential if you poison this pump? It'll become more positive, correct. Why is that?
Because you're no longer getting that one negative net charge inside.
Your concentration gradients are going to break down, right? Your concentration gradient, remember, what's determining your equilibrium potentials is the difference, the ratio. If your concentrations are equal, you got nothing. You got 1, basically. And so the pump basically keeps those gradients charged, if you will. The other important point to remember, and this will be more sort of obvious when we talk about action potentials, is that you're going to be using changes in permeability and changes in, of course, ion flux to generate changes in membrane potential. That's not going to involve the pump necessarily. Okay? You can fire an action potential. The number of ions needed to do that is very small. Okay? Tenths, hundredths, thousandths of a percent. And so you can poison the pump and still generate meaningful changes in membrane potential, things like action potentials. Okay? The pump generates the concentration gradients. Okay? It's a battery. Okay? You generate a stored potential difference, which you can do to do work. That's what a battery is. In the nervous system, the work is going to be information. Okay? The advantage of having a battery, instead of, say, drawing on this sort of directly for current, is you can activate a battery, you can draw on the current really, really rapidly. It allows you to pretty much instantaneously change your voltage. Close. That's very important for the nervous system, because you've got to operate the nervous system in real time with sub-millisecond precision in many cases. And so if you had to generate the current de novo using pumps all the time, that's probably not fast enough to generate the kinds of speeds that you want to have if you want to escape a predator or catch your food or whatever. So the pump is the ultimate source of the gradients. Okay? Ultimately it is. But it's the gradients that allow you to change your membrane potential rapidly. Okay? So the pump is the ultimate cause, if you will, but really the changes in gradients, the changes in permeability are the proximate cause. That's probably the best way to think about it. Okay? All right, let's talk a little bit about relative permeability at rest. Because again, that's going to be very important for determining the characteristics of the resting potential. So the Nernst equation itself really only gives us Vm for one type of ion channel. Okay? It only allows for a single ion to participate. And that's great if you're in a laboratory situation. Neurons have multiple types of channels. So what happens? Well, again, you could probably intuit it and say if the membrane were equally permeable to sodium and potassium, Vm would probably be some sort of average of the equilibrium potentials for potassium and sodium. And it turns out that's the case. In normal neurons at rest, the membrane is actually about 40 times more permeable to sodium than potassium, than sodium. Sorry. In this case, where we have multiple permeabilities for multiple ions, you use a derivation of the Nernst equation, referred to as the Goldman-Hodgkin-Katz equation. That basically is the same as the Nernst equation, but it accounts for relative permeabilities. So basically, the idea is that you use the same sort of basic term, that's RT over ZF times 2.303, log. And then instead of just having K over Ki, you have to basically multiply it by the relative permeability. So that would be, this would be 40 and this would be 1, because it's about 40 to 1. And so when you do that calculation, you figure it out, you come up with the resting potential value that we talked about, minus 65 millivolts. That is why the resting potential under normal circumstances is not the potassium equilibrium potential. There is a contribution of sodium. And again, remember, sodium wants the membrane to be at 123 millivolts, and potassium wants it to be at minus 90 millivolts. And basically, the resting potential is the weighted average of those two, weighted by relative permeability, approximately. I mean, with the log introduced into it and so on and so forth. Okay? Okay. This is the general form of the GHK equation. The only important point to note is, remember, if you have a negative ion, like chloride, you have to invert in versus out. Because remember, you have to factor z across into the logarithm function. And the way you do that, of course, is to flip the ratio. So for negative ions, it's not outside over inside, it's inside over outside. Why, I hear you cry, does chloride not contribute very much to the value of the resting potential that we're talking about? Yes, you were all wondering that, weren't you? Well, that's very easily answered. What is the equilibrium potential for chloride? What is the resting potential? Therefore, there's very little driving force on chloride at rest. There is actually plenty of chloride permeability at rest, but chloride does not contribute a great deal to the resting potential because its equilibrium potential is so close to the resting potential. Okay. As we will see, chloride is an important player for changing membrane potential under other circumstances, which we will talk about. All right, where were we? All right, so what happens to ions when membrane potential is changed? We actually basically already talked about this in terms of thinking about relative directions of flow in terms of really integrating this driving force equation. This is actually the exact same thing we were talking about. We already talked about it. So, any questions about that? The important point, again, is that if we're at the equilibrium potential, there's no net flux of a particular ion. As we move away from equilibrium potential, current will flow to attempt to bring the membrane potential back to that equilibrium potential. That is the important intuition for understanding ion flux. That will be very important when we think about action potential. Okay? Okay. All right, we're through 1, 2, and 3. Are any lingering questions about resting potential generation? All right, I just want to talk, again, I like to talk a little tiny bit about some clinical aspects of things that I talk about. Not that we talk about a lot, but I do want to talk about a couple of aspects of the sort of understanding of ions across membranes. A very important player in this whole process was Sidney Ringer. You've probably all heard of Ringer solution, which you give to patients in some kinds very large amounts. This is the guy who invented Ringer solution. It's named after him. So when you talk about Ringer solution, got to capitalize that R, because it's named after Sidney Ringer. Published 4 very important papers in the late 19th century, in the 1880s, studying the frog heart, actually. And basically, he found that a very simple mixture of ions, shown here, can basically sustain a frog heartbeat. You can take a frog heart out, and if you put it in the right solution, this, it will keep beating for quite a while if you keep it cool, 24 hours. Don't try that with your heart or a mammal heart, because it'll stop. And so the idea, he changed the various concentrations of these ions to try and understand the genesis of the heartbeat. As we know now, of course, the heartbeat is generated again by changes in membrane potential of muscle fibers within the heart, basically. And so, not surprisingly, his changes in these various ions had effects on the heart. But what he found was actually potassium had a much bigger effect than things like sodium. Changing potassium would basically change the refractoriness, basically the ability of the heart to beat, very readily for relatively small changes in concentration. And he found that in excess of potassium, the period of diminished excitability is increased and the frequency of heartbeats diminishes. Actually, if you raise the potassium concentration enough, the heart will stop. And again, that has to do significantly with the breakdown of its resting potential across the cell membranes in the heart. And so there's a rather somber application of this. And California is nominally a death penalty state. And it turns out that, do you ever wonder about that in terms of lethal injection, which is a typical way of stopping your heart, killing you? What is the agent that they use to stop your heart? Potassium chloride. Yeah, they just inject potassium chloride intravenously, and that's what kills you. All the other stuff they inject is basically to prevent you from suffering too much while that happens. So you're anesthetized usually with thiopental. You're paralyzed with things like pancuronium or tubocurare to stop muscle contractions. But it's the KCl that kills you, okay? Because it stops the heart. Ultimately, it would also stop your nervous system too, but it stops the heart more rapidly. And now you can understand why that works. Because if we have this if this is our resting potential generator, and we change the concentration of potassium, so, excuse me, by, let's say we change it. This is what we changed it to. This is our natural, minus 65. If we increase our potassium concentration outside, let's say to 95, of course that changes that ratio, changes your resting potential very drastically, as sodium now tends to dominate. Turns out when you do that, your sodium channels drop into what's referred to as a refractory state, and you can no longer fire useful things like action potentials, including cardiac action potentials. Stops your heart. So it's important to understand this stuff. Okay? You can actually have pathological states where ions are dysregulated, okay, in the plasma, which basically is the same as the extracellular fluid, interstitial fluid. As you might expect, this is something you do not want to mess around with very much. Okay? We just talked about the importance of that resting potential, and the importance, we'll reiterate it when we talk about action potentials. There are a whole variety of syndromes that will generate various changes in extracellular concentrations of ions, particularly syndromes involving, of course, the kidneys, which control much of the environment, if you will, of the plasma. So you can have all kinds of high and low concentrations of these various useful ions, particularly hyperkalemia, hypercalcemia, hypernatremia. You can also have the corresponding hypos. I'm not holding you responsible for this. Actually, it's really more for your interest than anything else. The point is, when you look at symptomology of these various things, they have multiple symptoms. Okay? They affect a lot of different physiological systems, but they're all going to affect, particularly potassium, things like cardiac arrhythmia. Potassium is probably the most dangerous, and we talked about why. Because at rest, it sets up that resting potential, and it's the most permeable. Hypercalcemia, again, can do a lot can actually cause a lot of symptoms, usually caused by hyperparathyroidism, for example, which of course keeps the calcium concentration set. Affects cardiac action potentials, calcium currents, which are going to be very important for a lot of things. Hypernatremia, sodium, again, affects membrane potential significantly. Also affects action potentials and synaptic potentials. And so again, it's going to affect the heart, the nervous system, and of course other physiological systems, because a lot of various sorts of pumps, as you've learned, rely on these ion gradients as well. So a lot of pumps may rely on, say, a sodium gradient or a proton gradient. You just regulate that gradient, there are all kinds of other things that will go wrong. Okay, so that's the clinical tidbit, I guess you might say, for ion regulation. As I say, we're not going to talk a lot about it, but it's important to think about. Hypoxia. Well, hypoxia is actually a fairly common problem. It just means insufficient oxygen to a particular tissue. It's not good for any kind of tissue, but it's particularly bad for nervous system tissue. And now we know why, or one of the main reasons why. Sodium-potassium ATPase uses an awful lot of energy to maintain membrane potential, and it needs oxygen to run. To generate large amounts of ATP, you got to have oxygen. Without ATP to run the pump, VM slowly depolarizes, which has effects like high potassium, which we just talked about. You die, or cells die in a localized area where you have hypoxia. Also affects many other energy-dependent pumps, leading to transporter failure, loss of volume regulation, so your cells explode, apoptosis, which is cell death of various sorts, cell lysis. So hypoxia generates a whole variety of events that blow up cells and kills them and prevents them from functioning normally. Turns out that's not the worst of it. Turns out that when you have hypoxia and you have uncontrolled cell lysis, the stuff that's inside the cell, the intracellular contents, spew out into the extracellular space. There's some very bad things inside cells, particularly glutamate, which is a neurotransmitter, and potassium. We already talked about why potassium is so harmful if you raise its concentrations outside of cells. Glutamate is similar in that it also tends to depolarize cells. Both these things can kill cells. So during a stroke, for example, which is a very common example of hypoxia of the brain, either because of a blocked vessel or because of a ruptured vessel, a hemorrhagic stroke, not only do you get death at the site where you have hypoxia, primary cell death, you get secondary cell death in the penumbra around it because of the crap that gets released from the intracellular space when cells die. So if you're interested in thinking about stroke treatment, one very sort of interesting avenue to think about is if you can prevent the secondary cell death, you can actually improve stroke outcome. And there is a lot of interest in trying to prevent or ameliorate that secondary cell death. There's usually not much you can do about the primary cell death. By the time you get the patient into the hospital, those cells are probably mostly dead. Not entirely, but mostly. Anyway. So again, thinking about membrane potential is very important for understanding at least some of the deleterious effects of things like hypoxia. Okay? Okay. Stroke and necrosis, I sort of talked about that. Direct cell death, necrosis, secondary cell death in the penumbra, caused by various factors. Yeah, I talked about that. Any questions? Yeah. All right. That is the bulk of that particular lecture. Where's my tech whiz? Oh, well. Guess I'll have to punt. So can I just close this? Or do I have to? Oh, well. A break? All right. 5 minutes or less. I'll find our guy.
I got mine first. Yeah. They're my cousins.
I was thinking, I could tear down that Thomas Gator wall when weirdly. It's like there is no Thomas Gator School of Medicine in the Valley. We're not in the program. They're not here. I'm just wondering. I'm just speculating. I'm being a hater. And you know, haters got hate. So I'm just doing my job. Haters gotta hate. So. Hater, oh, hate not haters. We're witty this morning, are we? What? Hospice, like usual. Yeah, it's this little lady that, you know, she's on hospice, so I hang out with her for like a couple hours. Chat with her about whatever. Actually, I just pretty much through the, I volunteer with the hospice. They assign me. They're like, okay, you're gonna go to this house. Okay, wait. And you just stay with that person until they pass. So they have, whatever that is. I'm interested in it. We'll see how it goes. I want to be involved in it. It's interesting working with girls. I mean, like, there's a song by Jane's Addiction where, like, it's like, you know, women, you know, people are like children, adults are all like children, adults.
You could have just showed me how to.
Like everyone, like,
Oh well.
like elderly are like children.
Therefore, since he's not here,
Children are there. It's just because, like, I asked her, like, why do you listen around, like, during World War II and the bombing program? She's like, I don't know. Out loud. So, yeah, she has really severe advanced dementia. So she thinks that, like, her family abandoned her for days on end. And I'm like, and you don't take issue with that, right? So it's like, you just validate what you're feeling. Feel her, because I actually like tried to edge in like maybe I remember differently, and she didn't like that at all. Like I backed up with that. She really believed that like her whole family abandoned her for 3 days straight. Which, I mean, maybe they did.
Yeah. Oh, sure, if you know where they live.
I'll be right back.
She's kindly getting the IT home. She would not tell me how to run this thing, of course. I'm obviously too stupid to do it.
Job security for him.
Yeah, there you go. But he neither did he reappear at 1 hour as he said. So we'll just, I suppose we will wait another minute or two. It's working. That doesn't mean it's recording. Well, we'll launch through and we'll record when we get to it. No problem. Just have to take better. Now, so you have to pay attention because we may or may not actually be recording. Ah, here we go. Ah, you're in luck. And I was just going to start. Sure, why not? It doesn't need to be. I figured I'd just keep going and hope it kept recording. Okay, well, that would have worked then. Thank you. All right, thanks. I'll probably just plow on then if I get to the next one. It's just easier. All right, we'll just plow on. All right, so last time we were talking about resting potential and the generation thereof based on really balances of ion and chemical gradients. And in thinking about the way the membrane potential is manipulated, in various forms, it's important to think about a particular category of channels, not the resting channels that we've been talking about, but gated channels, particularly voltage-gated channels. And these channels are going to turn out, of course, to be extremely important for generating this little beast here, and that is the action potential. Why is the action potential so important? Well, the action potential is unique to neurons, and neurons use it really for a very specific thing, and that is to carry information over relatively long distances. Relatively long distances doesn't mean miles, but it does mean, in some cases, a couple of meters, if it's an axon that extends, say, from your brain down your spinal cord, and you're a giraffe. But the point is that, as we'll see, the kinds of changes in membrane potential that you get without action potentials, so-called graded potentials, are really not very useful for carrying information long distances. Okay? They tend to decay rapidly with space. Action potential, because of the way it's generated by these different populations of voltage-gated channels, has some very important properties. One of the main ones is that it's propagated basically without any decrement. So very useful for sending information over long distances. So we're going to talk quite a bit about the action potential, how it's generated by voltage-gated channels, and again, how that generates useful properties that, again, the nervous system is going to use for manipulating information. So this is a basic oscilloscope trace of an action potential, which of course means this axis is time, blah. This axis is membrane potential. So here's resting potential. In an action potential, you go very rapidly from rest up to a particular potential, the overshoot potential, and then it actually returns to baseline, to rest, and actually undershoots baseline a little bit, typically, over the course of approximately a millisecond. So the time base here, this width, this from here to maybe here, it's just about a millisecond. So an action potential is a very rapid change in membrane potential that, again, lasts about a millisecond. The action potential is triggered by depolarization. Okay, depolarization is a jargon term. It just means making the membrane potential more positive. So as you go from, say, minus 65 at rest to minus 50, that is depolarization. The opposite of depolarization is hyperpolarization. That is making the potential more negative. So what that means or implies is that there's some sort of mechanism that is activated by depolarization. And the key to understanding the action potential and its characteristics is to first understand what happens to cells when they're depolarized and the kinds of channels, as we'll see, that are activated and inactivated by this process. A lot of this was done in a series of very famous experiments particularly by Hodgkin and Huxley in the 1940s, using the voltage clamp method, which is a physiological recording method that allows you to actually look at these kinds of currents directly in a very important way, for which they won the Nobel Prize. The Hodgkin-Huxley model of the action potential, considering it was formulated almost 70 years ago now, is still used. Okay, that's how good it is. The experiments that they did were remarkable experiments that were fundamental for our understanding of how the action potential and a lot of other membrane potentials work. So, all right, so Hodgkin and Huxley worked on a particular preparation, and that was the squid giant axon. If you take a squid apart, turns out they have a very large axon. It's used in the escape reflex, as you might expect. And the advantage of that is it allows you to record from that axon. It allows you to control and record the membrane potential and membrane currents very efficiently for reasons that I'm not going to go into. Also, you can get a lot of squid very easily, and you can eat it when you're finished with it. Hey, I used to work next to a lab that worked on lobsters. And they only used the eyes. So they had plenty of lobster left over. It was nice. Anyway, so they determined some very important fundamental things. They actually determined a lot of things, but they were very interested in the question of whether neuronal membranes have voltage-dependent changes in their permeability. Okay? Voltage-gated channels, ultimately. This is before channels were actually known. And more specifically, do ionic currents flow across the membrane when the voltage is changed? And is there a difference between a depolarizing epoch and a hyperpolarizing epoch? So they did some very simple experiments. I'm going to show a lot of these kinds of experiments, so it's important that you understand what we're showing here. In each of these cases, what we've got are usually two sets of traces. One, we're looking at membrane potential, okay? Minus 65, minus 130. It'll be labeled as VM, typically, or millivolts, something like that. And then this particular plot is showing corresponding membrane currents, expressed in this case as current per square centimeter. So it's actually current per square centimeter of membrane. That's not so important. The point is with voltage clamp, which is what we're talking about here, voltage clamp allows you to manipulate and control the membrane voltage. So we're going to be changing membrane voltage as one of our variables. And then we're going to be seeing what happens to current. Okay, so basically the outcome we're interested in is current. And the manipulation we're going to be doing are changes in voltage, which the voltage clamp allows you to do. Yeah, again, I don't want to talk about the technology of voltage clamp. So the point is we have sort of two very, at least in the surface, very similar seeming experiments here. In each case, we're just going to take the membrane potential, which starts at rest of minus 65, and we're going to just step it with a square pulse, either in a hyperpolarized direction by 65 millivolts or in a depolarized direction by 65 millivolts. So in each case, we're going to change the membrane potential 1 to minus 130, 1 to 0 millivolts. As you know from what we talked about last time, one thing that's going to do, of course, is change the driving force, right? But what's more interesting for the experiments for Hodgkin and Huxley is, does it change conductance as well? G. And the short answer is it can, as we will see. All right, so let's look at the hyperpolarization case, which is a fairly simple change. When you do your step, you get basically a very rapid, excuse me, very rapid inward current, you can see here, and which actually decays very rapidly back to baseline. That is actually pretty much exclusively capacitative current, having to do with charging and discharging the capacitance of the membrane. We'll talk some more about that when we talk about graded potentials. Now, there's something wrong with this picture. What is wrong with this picture right here? They've done something. They've cheated. They didn't tell you. I didn't tell you that they cheated, but they did. How did they cheat? What should we be seeing here in this, here, when we have a long square pulse of voltage?
All the way.
Shouldn't there be current here? Yeah, don't forget, based on our equation, driving force, in this case, you can assume it's potassium. But yeah, if you're changing the membrane potential in some continuous fashion, well, you've displaced it from the equilibrium potential, right? Current is going to flow, trying to bring you back to this resting level. The voltage clamp prevents you from doing that by injecting its own current. And so you should have continuous current, shouldn't you? Why don't we have continuous current here? Because it's been subtracted off of this case for clarity. If you actually did this experiment and didn't subtract off, they subtracted off the so-called leak current. That's the continuous current through the leak, which you determine based on a square pulse. They subtracted that off. They're only looking at currents that change. Continuous currents are basically filtered out or subtracted out. So you are correct. There should actually, what it should actually look like is something more like this, where you charge the capacitor, you have a continuous current, and then you discharge the capacitor back to rest, something like that. Anyway, it took me a while to figure out what was wrong with that. I kept looking at it. I'm like, this is not right. I've done this experiment, and this is not what I see. It's leak current subtracted, and that's going to be true of that's going to be true of the rest of the records I show you, which actually makes it much easier to look at the currents that are changing, which are the ones we're more interested in. But they did cheat a little. So, good spot in there, by the way. That's not easy. Not many people get that one. Took me a long time to figure it out myself. All right, so type of polarization, not very interesting. Really just passive current properties, nothing particularly unusual going on with conductance. Depolarization is quite different. It's similar in that you get your capacitative current at the start, but then you get a transient inward current and a delayed outward current, like this. Okay? It's exactly what you see. So, obviously, charge is in motion. You would think of it, by definition, as positive charge moving in, and then actually positive charge moving out. This very simple experiment, 70-odd years ago, establishes an important fact, and that there is voltage-dependent change in permeability. Okay? There's some kind of time-varying, depolarization-dependent change in the conductance. Okay? That may not seem like a very important finding, but at the time it was incredibly important. Okay? It was known that the action potential was triggered by depolarization. This kind of experiment is what ultimately allowed us to understand how does that occur on a molecular level through voltage-gated channels. Hodgkin and Huxley did not know there were voltage-gated channels. They actually generated the model that suggested that there were channels, okay? Which didn't get recorded until 30 or 40 years later. So anyway, any questions about that? All right, so because we can collapse the voltage, we can actually measure the equilibrium potential of these currents. So by stepping to different VMs, we can manipulate this driving force, and we can determine basically the direction of charge movement and the place at which there is no net charge movement, the so-called reversal potential. And so when you do that, if you take your membrane steps and you do a series of steps from, excuse me, stepping to minus 26, stepping to 0, stepping to plus 26, et cetera, you can look at the characteristics of these currents, and you can see basically when does a current go from, say, inward to outward. Because that's going to tell you that value. Why is it important to know this value? What very important fact will it tell you about the generation of this current?
How much it wants to get back to that point.
And what does that tell you about, again, what does that tell you about?
How quickly the ions will flow.
It actually doesn't tell you that. That had much more to do with the characteristics of this conductance, actually. But it tells you something very fundamental about this current. Okay? Well, what determines the equilibrium potential? The concentration of ions in a particular, so what's it going to tell you?
The forces at play between the two.
It's going to tell you the ions, right? Remember, we talked about the equilibrium potentials. You can calculate equilibrium potentials. They had a pretty good idea of what the equilibrium potentials for various ions were. So if you say the equilibrium potential is minus 90, you can pretty well assume that the bulk of the charge carrier is potassium, right? If the equilibrium potential is, say, 120, then you can assume most of the charge carrier is sodium. So by determining the equilibrium potential, you can determine something very fundamental about the current, and that is who is the charge carrier? Sodium, potassium, what? That will also tell you then, of course, what kinds of channels are involved. All right, so what did they find? Well, more to the point. When you step to, say, minus 26, you're still seeing inward current. We're only focusing on this inward current. We'll talk about this outward current a little later. And if you look at the inward current, what you can see is that it reverses at somewhere around, for a squid, about 50 millivolts, and actually is a little bit positive at 65 millivolts. So the point is its equilibrium potential is somewhere around ENA, excuse me. And so again, the Hodgkin and Huxley hypothesized or intuited that this was almost certainly sodium. Okay? That's one important thing. Another important thing is, again, just looking at the kinetics of this whole thing, which is a separate issue. One thing you can see, of course, is the inward current that we're talking about is relatively rapid, okay, and relatively short in duration. Okay. Then there's this outward current. Okay. This outward current, notice that we don't get the reversal potential explicitly for that. We didn't pass through it. So its reversal potential is somewhere below minus 26. It's potassium, of course, as you know. So it's about minus 80. And it's also much slower and much more prolonged. So again, we're looking both at the ionic bases of these particular currents, but also the kinetics, which of course is also going to be extremely important for understanding our rapidly changing dynamic action potential. Okay? One other interesting point, and that is if you look at just looking at the amplitude of this potential, this current, and just look at these three panels. What do you see for the amplitude of this particular current, the inward current, for just these three voltages? What does it look like? What does it do between here and here?
It's greatest at zero.
Right, it's greatest here. It's smaller here, it's bigger here, it's smaller here. In thinking about the amplitude of that current, okay, the relevant equation is this, okay, right? And we were just talking about driving force and the fact that, of course, ultimately the driving force determines the direction, but also the size, okay? The other factor is conductance. And the fact that this, remember, if the conductance stayed constant, let's say the conductance were constant across this here, would you see this increase? No, why not?
Because you need a change in that value of G to get a different.
We're assuming, well, but why? Okay, let's assume that this conductance is constant across these three panels. What would happen to the amplitude of this current if the conductance were constant? No, it would not stay the same. It's very important to think why not. No, it would not increase.
Or decrease then.
Increase or decrease, or well, you only have three choices. Increase, decrease, stay the same. Which one is it?
Oh, decrease.
Why? What's decreasing? Well, yeah, ultimately this is decreasing, driving force. If conductance were constant, you would see smaller amplitudes till you reach zero, and then of course you'd see opposite amplitudes. Correct. So the fact that this actually gets bigger tells you what about the conductance? It's getting bigger. Correct. Again, that's extremely important. That was in many ways the question we were trying to answer. Is there a change in conductance that is dependent on voltage, on depolarization? Yes, there is. That basically in and of itself answers that question at a very basic level. Doesn't tell you very much about it, but it tells you that it's there, okay? That there is an increase in conductance that is dependent on depolarization. More depolarization, to a point, gives you more conductance. Yeah? Why don't you actually see an action potential?
Oh, no, like, could you go back a slide? Like, on the right.
Where's the capacitative current?
Yeah.
Also electrically suppressed or filtered. Good call there, yeah. Once again, in this particular case, we're subtracting not just the leak current, but we're also ignoring the capacitative current, which ought to be there. Yeah, for emphasis on what's important. You're correct. If you actually did this experiment without subtraction of various currents, it wouldn't look exactly like this. It would be a lot more complicated. They've subtracted those out to enhance our ability to look at the active currents. Yeah, good call there too, yes. Yeah, there's a little subtraction going on here. Other questions? So, right, this very simple experiment tells us something very important. Some very important things about the current underlying the action potential. One, it's associated with an increase in conductance. That's very important. And secondly, it's almost certainly carried at least significantly by sodium. Okay, this inward current, that is. The outward current, no. We haven't talked so much about that. All right, so again, if you just plot this out, this is membrane potential here, this is the amount of current, you can generate basically a plot of current versus voltage. And if you plot both of these together, this is the early current amplitude here, this is the late current, what you find is that you can determine the reversal potential. So there's the reversal potential for the inward current, just about at ENA, about 50-odd millivolts. The reversal potential for potassium is down here somewhere. And actually, that's where you cross this line. So again, it suggests strongly that the inward phase is due to sodium flow. The outward phase is actually mostly potassium. It's going to turn out that's true. And that depolarization activates this conductance. That's why you have this dip. That tells you have voltage dependence, basically. Because as you depolarize, you get bigger currents because of the increase in conductance, basically. Okay? Pardon me. So if you're going to try and assert that this current is sodium dependent, there's a couple of confirmatory experiments you really ought to do. And Hodgkin and Huxley did that. And basically the idea was that in the first experiment we were manipulating Vm, okay, one component of driving force. In these experiments you can also manipulate the equilibrium potential. How do you manipulate equilibrium potential for sodium?
Add more ions.
Right, change the concentration gradient. Sodium replacement experiments. So for a squid actually has a even higher, being a sea mammal, listen, not a mammal, a sea organism. It actually has an extracellular sodium concentration that's very high. It's basically seawater. But you can manipulate that. You can remove sodium outside. That's going to make ENA more negative. And you can actually show that the direction of the early phase reversed from inward to outward. So you can actually basically flip the current, in this case, not by changing the the membrane potential, excuse me, but by changing the equilibrium potential. Okay? And then when you go back, wash your regular concentration of sodium in, you can actually show that you go back. So by removing the sodium outside, you can actually show that, again, this is the direction that is predicted by changes in ENA by the Nernst equation. It's very good evidence that the early phase is due to sodium. They did similar experiments looking at potassium, which actually, again, demonstrated that the late phase is not really affected by sodium, but affected by potassium. Okay, so it's a potassium selective conductance. So we have two voltage-dependent conductances. We have a rapid sodium-dependent conductance and a relatively slow, protracted potassium conductance. Okay? These are actually more experiments that were done. I actually forgot I had this in here. You can actually show, these are later experiments showing that you can use pharmacology to block out these various currents selectively. There are selective blockers for sodium, the sodium channels, the main one of which is, of course, tetrodotoxin. Tetrodotoxin is the toxin you find in your toxic sushi, fugu. That's why it's toxic. Tetrodotoxin is actually one of the more powerful neurotoxins around. Don't eat it. People still die of it every year. When you add tetrodotoxin, you block the sodium current, but not the potassium current. There are other blockers like tetraethylammonium that will block the potassium current, but not the sodium current. So you can do a variety of ion substitution blocker experiments to really show that the currents involved are sodium and potassium based. Okay? Yay, did I skip a slide? All right, I've already talked a lot about this. And again, in thinking about these currents and the direction of ion flux and the amount of ion flux that we see in a plot like we were just talking about, this is really the relevant equation. Okay. This equation is, if you got to remember one single equation, this one. This is the one that will allow you to intuit and allow you to determine things like direction of ion movement, identity of ions, and to a certain extent, changes in conductance, if you factor out things correctly. So, you can convert current to conductance fairly simply. And so Hodgkin and Huxley actually looked at that in terms, this is current, of course. These are the conductances, these are the associated conductances in millisiemens per centimeter squared. And basically, it shows pretty much exactly what you would think. If you look at the sodium conductance, so let's say you block the potassium conductance so it's easier to see, what you're going to see for, again, stepping to different voltages is it's voltage dependent. So if you go to a more depolarized voltage, you tend to get a larger conductance. But the conductance does saturate. Okay? It doesn't keep getting bigger. There's a certain point at which your conductance will saturate. So there's a limited value for that conductance change. And of course, the conductance is also very rapid. The scale down here is actually milliseconds. And so the sodium current is really completely over within about 2 milliseconds. So it's a transient voltage-activated current. Potassium current is similar in that it's also activated by depolarization. It also reaches actually a maximum conductance. But it's different because it's actually considerably slower to activate, and it actually lasts a long time. Okay? It actually lasts pretty much as long as your square pulse. Notice the sodium current doesn't last anywhere near as long as the triggering voltage, but potassium current basically does. Okay? So the kinetics of these conductances is very different. Okay? Both conductances activate faster for bigger voltage changes. So the bigger the voltage change, the faster the conductance activation. In fact, if you plot the conductance for various voltages, you come out with these kinds of activation curves. This is membrane potential. Again, this is conductance. Again, you can see that they're very steeply voltage dependent between about minus 30 millivolts for potassium and maybe minus, just about minus 30 millivolts for both of them. There's a very steep conductance change that then saturates out to the maximum conductance at about 20 or 30 millivolts. So has a very, very little conductance on these channels at rest, relatively high conductance over a pretty short interval of depolarization, maybe 20 or 30 millivolts. So they're very highly activated by depolarization. Okay? All right, so key points on that. Depolarization causes activation of a sodium conductance, activation of a potassium conductance. We showed that. It also causes an activation of a sodium conductance. Remember, sodium conductance is transient. Okay? It stops very quickly. Turns out that inactivation itself is also voltage dependent. It's going to turn out to be important for understanding certain aspects of the action potential. So not only is sodium channel activation voltage dependent, but so is its inactivation and its closure, but to a certain extent. Okay? All right, so if we're thinking about the voltage that I showed at the start, this is the action potential voltage. So again, it's this rapidly time-varying voltage. If we sort of plot that in conjunction with the underlying conductances, you can note some fairly important aspects of the action potential, again, based on the conductances. First off, if you look at the sodium conductance is really turning on very rapidly, it's peaking very rapidly, and it's really pretty much associated with the rising phase and the peak of that action potential. Okay? By the time you're getting back to the falling phase, if you will, the sodium current is pretty much done. The conductance is pretty much closed, and the potassium conductance is actually peaking, in the region of the falling phase, and actually, of course, in this undershoot as well. So it's pretty clear that the different parts of the action potential, the falling phase and the rising phase, are associated primarily with the activation of each channel. So the rising phase, the activation phase, is primarily associated with the sodium channel, sodium conductance. The falling phase is much more closely tied to the inactivation of the sodium channel and the subsequent activation of the potassium channel. Okay? And again, that's based on the kinetics and the properties of the channels themselves, which we'll talk a little bit about in a minute. Okay? All right, so we're going to return to sodium and potassium in the lectures 3 and 4, and a little bit more about how they give rise to action potentials. I'll talk a little bit about channels specifically. Hodgkin and Huxley actually predicted independent channels for sodium and potassium. At the time these experiments were done, there was certainly a lot of dispute about how charge got across the membrane. And one of the important hypotheses about that was that there were actual physical channels, molecular channels that punched through the membrane. These sorts of experiments, and actually other ones that Hodgkin and Huxley did, predicted these channels and predicted that they were independent, so separate entities for sodium and potassium, which turns out to be true. So I want to talk a little bit about these channels and about some actually later experiments that actually observed these channels directly, and a little bit about their properties. Okay? Because Hodgkin and Huxley actually in their original experiments didn't actually observe single channel events. That had to wait for, 40 years from that, late 70s and early 80s, with the advent developed by Nair and Sackman, the so-called patch clamp method. Patch clamp method is kind of cool in that what we've been talking about up till now is recording from the transmembrane events, voltages or currents, if you will, for a big chunk of membrane, for a whole cell actually in many cases. Here we're going to look at a very teeny tiny piece of membrane. We're going to take an electrode with a diameter of maybe a micron, 1 to 1.5 microns. And basically what you can do is you can seal your electrode onto the surface of a neuron. Turns out that the neuronal membrane is basically oppositely charged from the glass, and so they'll actually stick together very efficiently. When you do that, you can actually pull off a patch of membrane. You can actually pull the electrode back, applying a little suction, and you can record in basically two different modes of patch. You can have the inside face of the patch facing the extracellular fluid, so-called inside-out recording, or you can have outside-out recording, where you have the outside of the patch facing outside. And there's some technical tricks as to how you actually do it. Don't worry about whole cell recording. That's another way to record normal membrane potential. But the point is, you can have a very tiny piece of membrane with a diameter of maybe a micron. And it turns out when you do that, depending on the channel density of the membrane where you got the patch from, you may have only a single channel sitting in this patch. And you can actually look at single channel events, as shown here. Originally, this was done, I think the original experiments were mostly done for acetylcholine-gated channels, but for voltage-gated channels as well. So again, we're doing a similar experiment in voltage clamp that we were talking about before, where that we're stepping from resting potential of, say, -70 up to, say, 0 millivolts. That's going to activate our sodium and potassium channels. And when we only have this little tiny patch of membrane, we don't see these smooth, rapidly changing currents. We see very little discrete square currents. So a current that opens, that flows for a short period of time, less than a millisecond in some cases, and then closes, and then opens, and then closes. And the idea here is that if you have a channel that's gated by voltage, or say, in the closed state, you won't see anything. But with depolarization, you're going to increase the probability of opening. And with some probability, it's going to open. That allows current to pass. And the channel opens to a particular conductance state. Okay? It doesn't open a little bit and then a little bit more. Well, these channels don't. Channels can have a lot of very complicated kinetics, actually. But these channels are fairly simple in that they pretty much have a single open state under normal circumstances. And so you're either open like this or you're closed. So it gives you these little square single channel events. Okay. If you do that for a sodium channel, which I think I have in the next, yeah. So if you do that for sodium channels, you find something kind of interesting about the channel kinetics. That's actually shown here. So this is actually, each one of these traces is the result of a single step like this. So this experiment was done 1, 2, 3, 4, 5, 7 times. They actually did it thousands of times in the actual experiment. And as you can see, for each one of these depolarizations, you get a slightly different sort of pattern of channel opening. In many cases, you just get a single opening event and then silence. Some cases, you get a more complex opening event and then silence. Some cases, you get nothing. If you basically just average all these traces of individual channel opening events together over hundreds of trials, what you would get is a current that looks something like this. As you may recall, that pretty much, if you scaled it up, is basically exactly the same in terms of its kinetics as the macroscopic sodium current that we were just talking about. So it turns out you can regenerate the whole cell current that we just determined based on really just averaging up large numbers of single channel events. So what does that mean? Well, what it means is this depolarization event that we talked about, what does it do? Well, it basically increases the probability of sodium channels opening. That's one thing. But it only does it for a very limited amount of time. Where do I go? This is really annoying. I'm bringing my own pointer tomorrow. So it's really a statistical, the macroscopic current you see is really just a statistical representation of a whole bunch of really binary events practically. Yes, no. Did I open? I'm most likely to open very early on, and then I'm most likely to stay closed. Okay. That's what generates this basically rapidly rising, rapidly falling macroscopic current. This is a reflection of the probability of sodium channel opening, again based on membrane potential. And just as the conductance did, the probability of channel opening is also very steeply voltage dependent. In fact, it's actually practically the same curve. So you can really generate the current that you see based on just really the statistical properties of the single channel events. That's really kind of a remarkable finding. And actually, to be fair, Hodgkin and Huxley had predicted this based on a model, sort of really a conceptual model that there were these independent channel events, and they did have these sorts of properties. It wasn't until, as I say, the 70s when Nair and Sackman actually observed channel events and really showed that that was the case. Also quite remarkable experiments for which they win the Nobel Prize. So, questions? Yeah, okay. As you might expect, potassium channels are quite different from sodium channels. So again, if you take a similar step of voltage for a potassium channel, what you see again are single channel events. So channel openings, channel closings, but the potassium channel kinetics are very different. First off, the channel opening events are longer. They tend to be slightly delayed from the actual voltage step. So you see there's a little delay. They tend to be long. There's like openings and closings. And they don't really inactivate. They stay open in many cases with some fluctuations throughout the entire voltage step. Well, that's not surprising, because then when you average these kinds of single channel records to generate a macroscopic current, again, you basically show, or you basically generate the exact same kinetics you got for your macroscopically determined potassium current. So again, the characteristics of these channels basically allow you to determine probabilistically the characteristics of the macroscopic current. And that makes sense because the macroscopic current is generated by the sort of simultaneous activation of thousands and thousands of these individual channels expressed in the cell membrane at any given time. So again, these are very, very beautiful experiments that confirmed a hypothesis that, again, had been kicking around significantly since Hodgkin and Huxley, that we have these single channels, and their kinetics of opening and closing will basically allow us to construct the single channel, the macroscopic currents that we get. Okay, it's really quite remarkable. You can actually show currents through single molecules. They're very small, they're picoamps, but you can record them, remarkably enough. It's really quite a remarkable process. It's not even that difficult. I've done it, oddly enough, and it's quite doable. Any questions about that? Okay. All right, so there have been a great many studies on single channels, sodium channels, potassium channels, ligand-gated channels, all kinds of different channels. In some cases extremely detailed, especially now that we have the DNA sequences of many of these channels. They've been expressed in bacterial systems. Some of them have been crystallized. So it's interesting in thinking about these channels. This is actually data that, or sort of a structural model that Hodgkin and Huxley came up with. Sodium channels are subject to two processes, or two main processes, okay, regulated by depolarization. One is opening, activation. All right, we showed that. The other is inactivation. And so in thinking about that, Hodgkin and Huxley came up with the idea that a sodium channel has basically got two gates. Okay? One gate they call the M gate. That's the one that is actually opened by depolarization. And then there's actually another independent gate on the channel molecule itself they call the H gate that actually also, in this case, closes with depolarization. And the idea is that the M gate activation is rapid. Okay, so the M gates open rapidly, the channel opens, you get current. With a little bit of a delay, the H gates then close, and that inactivates the channel, prevents the current. Okay. And they actually generated an actually more formalized model that suggested the kinetics were basically M cubed. The M function was basically a cubic kinetic function. H was basically a linear function. That's based on data that we're not talking about. And for the independent potassium channel, the point is that there's only one kind of voltage-dependent gate. There's only an activation gate, which they call the N gate. Okay? And again, based on experiments, they made a hypothetical model about the kinetics of this N gate. And a great many experiments have been done trying to understand the kinetics of these various gates, these voltage-dependent gates, and we actually know quite a bit about them in terms both of their properties, in terms of responses to voltage steps, but you can also see them on things like X-ray crystallography. There's a lot known about it, which I'm not going to go into, because it turns out there are multiple open states, there are multiple inactivated states, there's a whole universe of complex kinetics, even for these relatively simple channels. that actually have very important roles to play in their functions, but that we're not going to talk about any more than that. So let's just think of them as these simple MH and N gates. That's, for our purposes, is sufficient. Okay? Yes?
Just for clarification, so we see two different kinds of gates. One because you see such a large increase in sodium at one point, and then the second because it's slower in activation, right?
Well, one gate, explicitly, when you look at a depolarization, you see two processes, basically. You see this rapid activation, but then out here you don't see any more channel openings. That's because the other gate is closed and you're in this inactivated state.
Okay.
Since that inactivated state is depolarization dependent, you don't actually remove the inactivation. You don't reopen that H gate until you repolarize back down to rest, because it's very steeply voltage dependent. So then when you relieve that inactivation, then you can reopen those channels, and they'll, of course, then will re-inactivate. So, does that answer your question?
Yeah.
Good. All right. I'll talk a little bit about ion channels. Ion channels are diverse, okay? That is the punchline. Hodgkin and Huxley talked a lot about the sodium channels and potassium channels of the squid giant axon. They analyzed them in great detail. This is extremely important. But there are a lot more types of voltage-gated channels than Hodgkin and Huxley knew about or could have predicted, okay? And I don't want to talk a huge amount about this, but I want to give you a little flavor for some aspects of the sodium and potassium channel diversity and why it's important for thinking about the firing properties of neurons. So for one thing, the notion that we were just talking about, that sodium channels always inactivate and potassium channels do not, is not true. It's true for squid giant axon channels. It's true for a lot of the channels you'd find in, say, your neurons, but it's not true for all of them. For example, the sodium channels associated with the heart are actually, most of them are non-inactivating. They're entirely different molecular species. They're sodium channels, but they don't have the same channel kinetics, and there are good reasons for that. This diversity in ion channel types is really important for generating distinct physiological responses of neurons. I think, yeah, okay. This is a complicated figure with a very simple punchline, and that is you can manipulate the firing pattern of action potentials in a given neuron by inserting different sorts of sodium and potassium and actually calcium channels as well. So again, you don't need to know this in detail. The point is that over here on the left, we're showing a certain subset of inward currents, sodium channels and potassium channels, sodium channels and calcium channels of various types. Over here are a variety of potassium currents that you find in neurons. And down here is actually showing the kinds of changes in firing pattern you can get by expressing different combinations of these voltage-activated channels in a particular neuron. So in this particular neuron, if you give it a square pulse of depolarization, what you get is a train of action potentials. This is very typical of things like neurons in your cortex, for example. But if you insert different sorts of channels, you can change this sort of regular repetitive firing. You can change the spacing of action potentials, so the frequency, by expressing different kinds of potassium currents, for example. You can delay firing. You can decrease the firing, so to a single spike. You can actually cause it to change its rate over the course of the train, so it gets slower, so-called accommodation. You can generate little bursts of action potentials. And so the point is, again, there's a lot of detail on this slide. I don't want you to know all these currents. I don't want you to know all these patterns. The point is you can change the pattern. For the exact same depolarization, the exact same stimulus, a neuron can generate very, very different outputs. And those different outputs are based significantly on what kinds of voltage-gated channels it expresses. Sodium, potassium, calcium being the main players. Okay, that's really the point. One other point from this, which is not quite so clear, not only do different types of neurons have these different firing patterns, but a single neuron can regulate its firing pattern. It doesn't always respond the same way to the same stimulus. That's actually what's shown down here. These results, this little burst versus this little train, that's from the same cell. It's actually from a thalamic cell in your thalamus. It can exist in different firing modes, depending on its modulatory state. For example, this burst state, that's just how it would probably fire if you're asleep, for example. When you're awake, it's going to fire more like this. just, that's a very extreme example. Point is that neurons can regulate their firing patterns based on other sorts of processes, particularly modulatory processes, which we'll talk more about when we talk about synapses, okay? So not only is there diversity between different neurons, a single neuron by itself can generate diversity of its firing patterns. And again, it's based on the expression of these voltage-gated channels and how they're modulated by other things, which we're going to talk almost nothing about. But it's useful for you to know that, because actually when we talk about pharmacology, and you guys talk about pharmacological agents, some of the pharmacological agents you'll talk about, of course, specifically interact with different subtypes of, say, potassium currents versus others, or calcium currents. I mean, you've probably all heard of heart patients who are on calcium channel blockers, right? Well, again, it's blocking a very particular subtype of calcium channel that is very important for regulating heart rhythmicity. Now you know why. Again, it's generating cardiac action potentials. You can manipulate that based on modulation of the underlying molecular channels, for example. Okay? Now, this is really the same thing, just showing, this is actually a sagittal section through a rat brain, and it's just showing recordings from different neurons in different locations, just showing diverse firing patterns. It's just kind of cool to see, you know, sort of train firing neurons, burst firing neurons, weird firing neurons. Those are Purkinje cells of the cerebellum. They have really complex firing patterns, remarkably complex, because again, of the currents they express. And the actual details of these firing patterns tell you a lot about their function. And so, but that's all I want to say about it. I just like to show that because it's kind of cool. Okay. All right, so action potentials in drugs. Well, you know, if you're thinking about voltage-gated channels, voltage-gated currents, it's important to think about how you might manipulate them. And one of the ways we manipulate them, of course, is using various kinds of drugs. And there's really a couple of important things to think about, a couple of important reasons why drugs and ion channels are so important. First off is the ability to manipulate, to block these channels or to activate them with drugs has been really important from a research standpoint for our basic understanding of these channels. But secondly, in the clinic, some of these drugs are of clinical relevance. So I just want to, this is not a pharmacology class, so we're not going to talk in detail about all these sorts of agents, but I just want to give you a little flavor for thinking about agents that interact with these voltage-gated channels. And then when we talk about synaptic events, that's where pharmacology becomes really important. Most of the sort of psychologically active drugs you might give to a patient, most of those act at synapses in some way. Actually, very few of them act at sodium channels or potassium channels, but there are some. So let's talk about some of these. One very famous drug that interacts with the voltage-gated sodium channel that we're talking about is tetrodotoxin. Tetrodotoxin is famous, probably most famous, because it's the active ingredient in your typical pufferfish sushi, fugu. There's the pufferfish right there. Has anybody ever had fugu sushi? Anybody?
I know someone who has.
You do? I'm surprised. You can't really get it in the States, and it's really expensive. Well, the reason that it's really expensive is because in Japan, a sushi chef who does fugu has to be very extensively trained. The reason they have to be extensively trained is because my understanding of this, and I'm not sure about it, but part of the allure of this particular kind of sushi is this kind of numbness and tingling you get in your mouth when you eat it. That's tetrodotoxin blocking sodium channels in your mouth and causing numbness. Now, if that numbness should start to spread from your mouth into the rest of your body, you better call the paramedics, because it's blocking sodium channels throughout your body, and you will die. On the plus side, tetrodotoxin does not cross the blood-brain barrier, so it doesn't kill you by shutting your brain down. There wouldn't be anything you could do about that. It kills you by paralyzing your diaphragm, basically. So if you get on artificial respiration for a while, you can make new sodium channels, and you can live through a bout of tetrodotoxin poisoning. But every year in Japan, there are quite a number of tetrodotoxin poisonings. Usually people who aren't being careful and get some of the liver along with their meat, because the liver has very high concentrations of TTX. Anyway, some other critters with TTX, those are California newts. Don't eat them. They live around here. Blue-ringed octopus, very famous octopus, lives near Australia. That's why they're so dangerous. That's poison arrow frog. As I say, tetrodotoxin basically doesn't have any clinical use. unless you want to kill somebody in a mystery novel. It's very effective for that. But it's been extremely useful in understanding and characterizing the sodium channel. In fact, the original biochemical studies that allowed us to isolate sodium channels biochemically relied on the incredibly powerful interaction of tetrodotoxin with the sodium channel. As I said before, tetrodotoxin is one of the most potent neurotoxins we know. It's pretty much 1-2 with botulinus toxin, which actually acts at synapses. So it's been a very useful molecule for research and for food. But it's not something you're going to be prescribing to patients, thank goodness, in any clinical setting. Okay? So there are actual sodium channel blockers that are clinically important, as you might expect. And those are the so-called cane drugs, local anesthetics, Novocaine, Procaine, lidocaine, bupivacaine, cocaine, all of those guys. They're all also sodium channel blockers. Okay? They act in a very different way. They actually get into the channel and block the pore itself. So they're so-called open channel blockers. As you know, these are all very important as local anesthetic substances. You inject them, say, subcutaneously or into the nerve in your mouth at the dentist. You've all had that. And it blocks the nerve from transmitting impulses in either direction. So on the plus side, you don't feel any pain. On the minus side, you can't control your muscles. Again, so these aren't, one thing that's interesting to me, which I didn't know until recently, is they actually use local anesthetics intravenously for certain types of cardiac patients. They inject lidocaine. I actually would have thought that was a very bad idea, but apparently the cane drugs don't really cross the blood-brain barrier either, and it actually improves cardiac function in certain cases, which is kind of interesting and scary, but let's see. Yeah, that's the structure. Don't worry about it. They all have a very similar ring, organic molecule. They're all lipid soluble. Toxins of various sorts live in all kinds of different places. Okay. By a toxin, I'm really referring to a molecule in this case that binds up to one of these ion channels. These are ion channel toxins. And again, they've been very useful primarily for research. So this is actually a toxin fraction from a scorpion, Laris quinquestriatus, called the Israeli death scorpion or death stalker. And as you might guess from the name, it is not one you want to mess with. Its venom is quite toxic and also supposedly incredibly painful. Usually adults don't die from it, but children and the elderly do. The point is, and I don't want to talk a lot about it, the venom contains a whole bunch of different toxins. That's typical for venoms. They all do all kinds of different things. Slows sodium channel inactivation, blocks calcium-activated potassium channels, blocks the potassium channel called the delayed rectifier, blocks chloride channels, probably other things we don't even know about. It's all a cocktail designed to kill and paralyze its prey and its defense. But the point is, just getting back to the characteristics of sodium channels we were talking about, we were just talking about agents that block the sodium channel, right, and prevent it from acting. This particular toxin from the scorpion doesn't block the current. It actually enhances it. It blocks inactivation. So it actually, as you can see, normally your current falls off very rapidly. Here you can see it falls off very slowly. So it doesn't cause blockage of the sodium channel. It actually causes hyperexcitability, enhanced ability for action potentials to be fired. That's what generates things like pain, because your pain receptors are firing spontaneously, for example. It's also very bad for the various, we talked about rhythmicity of action potentials. Well, if you're messing with an activation, you're messing with rhythmicity of action potentials, which is bad. Cardiac rhythmicity, all kinds of other things. So anyway, the point is that you can manipulate these channels in negative ways with blockers. In many cases also, though, you can manipulate their kinetics in other ways that will have very different effects. And this is just a good example of that. Okay. Potassium channel blockers, there are bunches. There are actually bunches of different potassium channels. They have different sorts of blockers. In terms of blockers of potassium channels, the dominant effect is hyperexcitation. Convulsions would not be unusual. They all basically enhance action potentials in various ways. Things like 4-aminopyridine, which is causes broadening of spikes. Apomin, which is a major component of bee venom. It's one of the reasons it hurts so much. Again, it causes spontaneous firing of pain receptors in your skin, for example. It also does that for a lot of other reasons, but one of them is a potassium channel. So again, there are a lot of blockers and manipulators of potassium channels that are also going to affect excitability. Okay. A blocker of a potassium channel is generally going to enhance excitability. An enhancer of a potassium channel is generally going to decrease excitability. There's a lot of variance there. So, oh yeah, I forgot I had this in here. Not surprisingly, since channels are molecular entities, they're subject to mutation. And there are familial and non-familial mutations of genes that affect these channels in various ways that actually play out in various sorts of syndromes that you'll find in patients in some cases. This is a nice example actually from the veterinary world. Well, nice maybe isn't the right word. It's a syndrome that affects horses, mostly quarter horses. And it actually affects a very large population of thoroughbred quarter horses in the United States. The reason for that is it all comes back to a single quarter horse ancestor called Impressive. Mr. Impressive was impressive for a couple of reasons. One, they had very large muscles. He did, and so does his progeny. And they win a lot of races with these large muscles. That's not his procreatory accomplishments, although those were pretty impressive too. A lot of quarter horses are descended from him, somewhere around 55,000. However, they're also subject to this periodic paralysis, particularly if they eat something with too much potassium. They may become paralyzed, like this, or in some cases after exercise, like racing. Turns out that the whole process, that this includes both his high musculature, but also the paralysis, is because of a single mutation in a sodium channel. It's an autosomal dominant mutation in actually a muscle-specific sodium channel. There are a lot of different kinds. And it reduces inactivation. So just like we talked about for that scorpion venom, it decreases, one of the things it does is decrease the inactivation of the sodium channel, leading to hyperexcitability, which actually leads to, when you excessively depolarize and excite a neuron, basically it ends up in you get this hyperactivation like this for a short while, but then you get inactivation. Remember, that's depolarization dependent. That's where the paralysis comes in, okay? And for reasons that I'm not going to go into, and which in not all cases are totally clear, high levels of potassium actually enhance this process, probably by providing a little bit of depolarization that kicks off this hyperexcitability. The actual explanation is quite complicated, but it's a very interesting process. Similar mutations occur in humans. The human mutation is usually in the so-called SCN4 gene, which is a sodium channel found at the neuromuscular junction, which is the junction between motor neurons and muscles. And they show a similar sort of potassium-based periodic paralysis. So the point here from this is that there are quite a number of different sorts of syndromes that you would call channelopathies, based on either inherited or spontaneous mutations of various sorts of voltage-gated and ligand-gated channels. These are just some examples for your edification. You're not responsible for this. It's just kind of cool. Not surprisingly, the numbers of these are disorders of excitability, particularly types of inherited epilepsy, which is associated with, in many cases, sodium channel mutations or mutations of inhibitory systems like GABA. Probably one of the more common ones is cystic fibrosis, which you've probably heard of, which is a chloride channel mutation, for example. So they're, again, channels are used for a lot of other things. Migraines, pain syndromes, probably psychiatric disorders. So it's, again, a very interesting target for clinical intervention, at least impossible. So who knows? You may see them in the clinic someday. I guess most of these are actually pretty uncommon, is my understanding. But still, questions? All right. I guess we'll just zoom along. It's apparently recording appropriately. All right, so action potentials. All right, so we've talked about resting potential in terms of setting up this battery And the main purpose of this battery is to allow us to change membrane potential rapidly, like action potentials. And we just talked about the generation of action potentials based on these populations of voltage-dependent channels. What I want to talk about now for actually a couple of lectures mainly are properties of the action potential. What do the properties that we talked about for these single channels, what do those sorts of channel properties yield in terms of properties of the action potential itself? And why is that important? What is it useful for? Why do we have action potentials? What do we do with action potentials? What are their limitations? So talk a lot about action potential characteristics. We'll also talk about how those characteristics then allow action potentials to be propagated over long distances, which is really the whole point. Okay. So in thinking about action potentials, we're going to talk really about sort of two pieces of this. One is just properties of the action potentials, ups and downs, how it goes up, how it comes down, and how do the properties of the channels allow that to happen. And then we'll talk a little bit more about it in terms of theory, in terms of currents, conductances, a little bit more, and in and out. By that we're meaning current mostly. All right, so action potentials themselves, as we talked about, now we're talking not about voltage clamp and measuring currents. Now we're mostly talking about the actual potentials themselves, which are what you would see naturally, if you will. And in terms of recording those, of course, what you're going to do is record from the inside of a cell, either an axon like this, or a neuron, or a muscle cell. A lot of the pioneering work on action potentials was done on squid giant axons. A lot of other studies have been done on smaller neurons, cortical neurons, hippocampal neurons, muscle fibers. And, you know, action potentials are very diverse, but their sort of basic characteristics are fairly similar. And so, again, the idea is to measure this time-varying change in membrane potential. Okay? All this stuff is detail. You don't have to worry about this stuff. Yeah. One interesting thing is stretching the muscle will generate an action potential because there are actually stretch-activated channels, mechanically gated channels. Anyway, so the point is, what we're really interested in are the phases of the action potential and how the characteristics of the voltage-gated channels we talked about account for the kinetics of this potential. So we're really going to talk about the rising phase here, the falling phase, and then this undershoot. So the different kinetic, if you will, phases. And again, we'll talk about how that's generated and, again, some aspects of properties of the action potential that fall out of these various properties. So rising phase, overshoot, falling phase, undershoot. About a 1 to 2 millisecond event. Very rapid. All right, some very useful and interesting properties of the action potential. And again, we'll talk about the genesis of these in a sec. Of course, the main one we already talked about, triggered by depolarization. Okay, so that is how we are generating the action potential. Some sort of depolarization. In an experiment, we've been using square pulses of depolarization. Where does natural depolarization come from? In an animal, you don't have electrodes giving square pulses of depolarization. Where does depolarization come from in a normal animal cell? Neurotransmitters. A lot of them come from synapses. Yep, those are the dominant ones you would think of for your central nervous system. Anything else? There is. I'm thinking of a big one. Sensory receptors of various sorts, correct. Generator potentials, as they are usually called. Whether it's potentials generated by light hitting the retina, or mechanically gated channels in your skin, mechanically gated channels, hair cells in the ear, chemically gated channels in your nose. Those all generate depolarizations ultimately that cause action potentials. Although for photoreceptors, it's all backwards, but you don't have to go into that. So it's depolarization, correct. In order to get an action potential, you have to reach a potential called threshold. So if you have a very small depolarization below threshold, you're not going to get an action potential. You have to get a certain amount of depolarization. So the depolarization required for threshold is highly variable. Different cells have different thresholds. And again, an individual cell can regulate its threshold in a variety of different ways. It's usually somewhere around 10 to 20 millivolts from rest. So it's not a huge depolarization. It's not that big. Action potentials are all or none events. What does that mean? The amplitude of the action potential is basically the same. Every time you trigger it in a particular neuron, the size of it is pretty much the same. It doesn't matter how big the stimulus, how big the depolarization was. If you cross threshold, you get an action potential. If you don't cross threshold, you don't. So they're basically binary, if you will. They're yes-no events. It's actually very important for understanding how they propagate and how you might use them for coding, which we'll talk a little tiny bit about. Therefore, action potentials propagate without decrement. So they keep their amplitude the same. They move relatively slowly. Along unmyelinated axons, you probably run up to about 30 meters per second. Myelinated axons can actually run up to more like 120, 150 meters per second, which is fast enough for our purposes. Okay, and it's important that they be fast. You got to move information around your body quickly. The bigger your body is, the faster you got to go. At the peak of the action potential, inside becomes positive. That's the overshoot with respect to the outside. So that's the opposite of at rest. After an action potential, there is a brief set of refractory periods. There is the so-called absolute refractory period. That is the time during which another action potential cannot be triggered under any circumstances. The absolute refractory period is about 1 millisecond. Okay. Why is that important? Well, for a variety of reasons. One is it sets a limit on the firing frequency. How many action potentials can you generate per second? The absolute limit is about 1 kilohertz. Actually, very few neurons fired anything close to that rate, more like 200 to 500 hertz tops. So as we will see, that refractory period is also important for propagation. All right, so for the remainder of this little lecture, we'll see a little bit about why these properties arise, again, based on the molecular aspects of the channels involved. Yes, sorry.
I was under the understanding that absolute refractory period was during the action potential, and the relative was immediately following the action potential?
Well, to a certain extent, that's semantics, in that the absolute refractory period, you're right, it's like there is overlap of the absolute refractory period with the falling phase of the action potential. So you can think of it as being during the action potential as an entire unit. What I'm really talking about is time after the peak, primarily. You're right, I mean, that you can think of it as actually occurring, and then we didn't talk about the relative refractory period. That's a period of time during which it's more difficult to fire an action potential, as you obviously know. That's significantly associated with this undershoot, actually, and inactivation. So you're correct in that it's really semantics about after versus during. You're right. It's all continuous. And really the proper way to think about it is probabilistically, in that it's all about the effective sodium channel density, right? At rest, you have pretty much all of your sodium channels are available, so you can activate them and get an action potential very easily. But as you activate more and more of them, you have more and more depolarization, more and more of them are in the inactivated state. And so during that time, up here at the peak, you're pretty much 100% inactivated. As you start dropping back down, the probability of that inactivation decreases. So it's a probabilistic function. So there's a certain probability that you could fire an action potential starting probably about here, where you get to 50% availability. I'm actually not sure what the data is. And so that's how it's a continuum. It's not a sort of a hard and fast thing. And it's all based on the kinetics of the channels, which we'll talk about in a sec again. Other questions? Yeah, it's all probability. It's all single channel stuff is really what it boils down to. An experiment. All right, so this is actually really trying to think more about the characteristics of potential changes across the membrane. And really sort of contrasting, if you will, action potentials and potentials that aren't action potentials, so-called graded potentials, subthreshold potentials. And so the idea here is to look at an axon. So this is a sensory nerve fiber, an axon. And you're going to have two electrodes, one at a point A, one at a point B. And you're going to stimulate at point A, and you're going to see what happens at point B. So basically what we're interested in is propagation of potentials along this little length, which is a, a few centimeters. So a fairly short length. And so the interesting point is we're going to really compare and contrast what happens for potentials that don't trigger an action potential versus potentials that do. That's the main comparison we're going to do. So in this particular case, when we take our electrode at A and inject a little hyperpolarization, so this is our injected charge right here. This is the resulting voltage. When we look at B, we don't detect this change at all. Okay. Why is that? Well, again, it's because we generated current at this point source. As the current flows along the axon, it flows out. Okay? It flows out along the across the membrane resistance progressively with distance. So the further it goes, the more it flows back out of the cell. And so by the time you reach this point, effectively you have no change in membrane potential. Okay? You see the same thing for a small depolarization. Okay? A little tiny depolarization, you see a little tiny positive current, positive voltage, excuse me, that's a voltage. No change detectable at B. If B is close enough, you will detect something. You'll actually detect a smaller representation of this. Now, let us say at this point we give a bigger depolarization, which generates an action potential at this point. Now at B, we see that action potential. Okay. Notice something interesting about the action potential, though. What we don't see is this little subthreshold part of the depolarization. All we see is the pure action potential. But the point is that the action potential, as we were talking about, is propagated along the axon with a high degree of fidelity. That is in contrast to these subthreshold events, these graded events, which decay fairly rapidly over space and time. Okay. Therefore, if you're trying to move information over a long distance, like an axon, you need action potentials. That's what action potentials are for, long distance carrying of information. For just short distances, you don't actually need action potentials. And actually, in some ways, these subthreshold potentials are actually more efficient. They carry more information, which we'll talk about. But for long distances, you need this the regenerating action potential. Okay, that's what this experiment is telling us. Okay? All right, so we learned a little bit about the GHK equation today. And when thinking about the sort of overall sort of mechanisms underlying this complex dynamic membrane potential change, the GHK equation is a good way to be thinking about that. Because as we know, what we see as the action potential is all based on changes in permeability, right? It's all based on openings and closings of channels, okay, which is effectively changes in permeability. So at any point on the action potential, you could use the GHK equation to basically determine the relative permeabilities based on the change in membrane potential. Okay? Of course, it's a dynamic process. It's constantly changing, but you could. Okay? So if you think about it sort of very, very simplistically, I guess you might say, is that if we're going to depolarize the cell, which is what we're doing, the Vm is given by the GHK equation, and it's close to Ek. Okay? And it's close to Ek at rest because the permeability of potassium is so high. Okay? So the ratio of PNA to PK, that's what's shown here. It's just the ratio of sodium to potassium permeability. At rest, the ratio is low. Okay? When you depolarize, of course, the ratio is high, right? Because the sodium channels open, at least at the start, and that generates sodium permeability. So, of course, that's going to depolarize the cell, right? Right. So a large increase in the permeability to sodium occurs during the rising phase of the action potential. We showed a conductance plot of that. And so again, if you just look at and think about the GHK equation, that is going to basically predict what's going on at any point based on these, again, dynamic changes in permeability. Don't forget that the potassium permeability is also going to be changing during this time. And so it's really the coordinated changes in these two permeabilities that accounts for the shape of the action potential and most of the properties of the action potential. Okay? Oops. So again, just to remind you, I guess since it was today, it's not so important to remind you too much. But again, these are the underlying conductances. So these are the changes in permeability that we're talking about. Permeability and conductance are effectively similar. And so It's these at any given point that are going to tell you what voltage you see based on, again, the GHK equation. Okay? All right, so the all or none law, if you will, the all or none characteristics of the action potential, again, really rest on thinking about primarily sodium channels. So let's think about this rising phase and this overshoot, because those are really dominated by the properties of sodium channels. And again, you would guess that based on this plot, because of course the sodium conductance is the one that's rapid, and it's also over with rapidly as compared to the potassium conductance. So what happens? Well, at rest down here, very few sodium channels are open. Permeability is very low. If you stay at rest, that will continue forever. But if you start to depolarize the cell by your electrode or by a synapse or what have you, voltage-activated sodium channels open. We know that. We've seen that. Well, what happens when you open the sodium channels? The permeability to sodium goes up. Sodium rushes into the cell, carrying its positive charge. Therefore, the cell tends to depolarize more. Since it depolarized the cell further, more sodium channels open. That gives you more depolarization, gives you more sodium influx, gives you more depolarization. So it's this positive feedback process. Okay? The depolarization feeds on itself and generates itself, basically, by this positive feedback process. And so, excuse me, at GNA, the permeability, the conductance for sodium continues to increase, basically until all the sodium channels are open. That's a relative term, but let's not worry too much about that. One interesting point, other than sort of rest in the overshoot, is threshold. It turns out at threshold, the two currents are equivalent. INA equals IK. So there's a balancing point where basically the influx of potassium, the efflux of potassium is trying to keep the cell from depolarizing more, and the influx of sodium is trying to depolarize it more. At threshold, that's perfectly balanced. Okay? If you go one molecule of sodium over, you will trigger the action potential, because it's this positive feedback phenomenon. That's what positive feedback phenomena are good for. They're explosive. If you get past the trigger point, it will proceed to completion. And that's what an action potential does, very efficiently. Okay. So the rising phase, the activation and the sort of overshoot kinetics are really dominated by the sodium channel and its kinetics. All right? All right, well, we already talked about explosiveness. Let's talk about repolarization. Because, of course, it wouldn't do you very much good if the sodium permeability stayed high. What would happen if the sodium permeability stayed high? If you activated your sodium channels and they stayed open, what would happen?
It would flow to the first equilibrium.
And what would the new equilibrium be?
58 mil.
Yeah, you'd go up and you'd stay up. You would never come back down. And that wouldn't be very useful if you're trying to use action potentials to carry discrete bits of information. You got to bring it back down, and you got to bring it back down quickly so that you can have repetitive action potential firing. It's very important. So what accounts for this falling phase? Well, again, it's really a two-phase process. Very important component, of course, is that the sodium channels, when they're depolarized, not only do they open, but then they inactivate, which is a closed state. So as you're depolarizing and activating channels are activating, they're allowing current through, then they're inactivating. And they stay inactivated until they get back down to hyperpolarized levels. So at this point, sodium channels are inactivating, and actually most of them are inactivating. Therefore, and of course the driving force on sodium is decreasing, so you reach, that's your peak. Your peak is close to ENA. It's not exactly ENA because there is, of course, potassium permeability as well. But you start to fall back down, okay? And that's because, of course, your sodium permeability is dropping back down to its original levels. Close to zero, not exactly zero. Okay, so that's not enough, it turns out. Sodium channel inactivation, if that's all that you had, you would come back down to resting potential, so you would reset, but it would take you a lot longer. If you block sodium channel inactivation, you return to baseline on the order of tens of milliseconds, not 1 millisecond or less. So there's another process, of course, that allows you to repolarize very rapidly. That's the relatively slow opening of the potassium channels. So Potassium channels, of course, open with a little delay, and they actually stay open. They don't inactivate. And so potassium channels, of course, when they're open. Now, if you open a potassium channel at rest, what sort of net potassium flux do you get by definition? At rest. By definition,
Whatever direction.
it's zero net flux, right? Zero net flux, equilibrium state. I would like to see some nods. That's the whole point. We defined resting potential at the point of zero net flux, remember? Yeah, we did. If you we're opening potassium channels to repolarize this action potential. What would happen if you opened all those channels at rest to the membrane potential? It actually would hyperpolarize, you're right.
Those are leak channels, right? Arrest.
Yeah, you're right. I actually slipped up on that one because we're actually going to be explaining this by an increase in permeability. See, no one's perfect. Those of you who said potassium efflux are correct. Sorry about that, guys. You're right, actually. All right, well, never mind that. We'll come back and talk about that when I have my act together. More to the point in terms of the falling phase. When you open these potassium channels, what happens to potassium ions when we're up here? Okay, that's the more important question.
They rush out.
Right. Okay. And why is that? Because, of course, we're very far away from the potassium equilibrium potential, which is way down here somewhere. So there's a very strong force on potassium ions, excuse me, driving them out of the cell. And, of course, when you drive potassium out of the cell, that hyperpolarizes the cell very rapidly because you're carrying positive charge out. So the rapidity of the falling phase is really accounted for by that potassium current. And then the undershoot, sorry guys, this is because the permeability of potassium at this point is greater than it was at rest. Sorry, you guys were correct, I was wrong. At rest you have a certain permeability. Here you have that resting permeability plus the permeability associated with the voltage gated channels, or some significant fraction of them. That's going to drive the potential here closer to the potassium equilibrium potential, which hyperpolarizes it. Sorry guys, you were right. I meant to say that. Still, the potassium channels, of course, will then also close, which will bring you back to your original permeability state at rest. Okay, was that comprehensible except for the part? I know, I got a little part wrong, but I got corrected, so it's okay. So it's really the properties, again, the molecular properties of the channels that we talked about last time account for the kinetics of the action potential. Okay? And again, those kinetics are going to relate to very important properties of the action potential as well. Okay? Do I need to repeat that? Good, because I don't think I can. I probably could. All right, so this is really just a sort of a more detailed summary diagram of this idea during the repolarization phase of this sort of two-phase process. So remember, sodium channels, getting back to the channels involved, are dually gated. At rest, the M gate, which is the one that actually is activated to open the channel, the M gate is closed. Your sodium conductance is very low. At rest, the H gate is open. Both gates respond to voltage, but in different directions and with different speeds. M gate's rapidly open. The H gate slowly closes. And so again, you're going to get ion flux through basically because it's open, and then closure of the H gate and inactivation. Okay. So you have transient opening of sodium channels, and they stay inactivated even in the face of continuous depolarization. Okay. That will decrease the sodium permeability and bring ultimately you back down to rest. Although, as we talked about, it's too slow. So in order to make it fast, you have potassium channels. And again, the potassium channels are singly gated, just these N gates. The N gate opens slowly with depolarization. So it doesn't participate in the rising phase very much. But by the time you're getting to the peak of the action potential and a little past the peak, the potassium conductance is maximal. And then the potassium, potassium efflux then brings the membrane potential back to rest. And actually, because of the increased permeability, you actually undershoot rest a little bit because you go closer to the potassium equilibrium potential, which is about minus 85, minus 90 millivolts. Okay? So it's all about channels. At heart, it's all about molecules. What's important for thinking about it phenomenologically, though, is really the action potential, okay? The potential itself. That's what cells show, this. The molecular aspects of it that we just talked about, that's how it's done. So it's very interesting. All right, so in thinking about the refractory period, getting back sort of to her question, the refractory period, the period during which you can't fire another action potential, reflects this inactivation. Okay, since you have sodium channel inactivation at this point, where pretty much all of your channels are in the inactivated state, closed, you don't have a high enough channel density to support an action potential at all. So you can't trigger another action potential. Over time, of course, as you get hyperpolarized, channels are going to exit the inactivated state and become available. And of course, this undershoot is also going to decay. And so there'll also be a period of time during which it will be harder to trigger a second action potential, not impossible, because you have fewer sodium channels available, and actually the potassium conductance will also inhibit you from depolarizing. So that's how the refractory periods come about. Okay? It's not quite new. This is really, again, a very highly schematized summary of it in terms of thinking about it in terms of both the positive cycle, the sodium channel cycle, and the negative cycle. So the positive cycle, of course, all triggered off by depolarization, has to do with sodium channel opening, increased sodium current, which of course depolarizes the membrane more, generates more opening of sodium channels. The negative cycle part of course, is the potassium component, opens potassium channels, increases potassium current, tends to hyperpolarize. So it's really a sort of a two-phase process. Don't forget sodium channel inactivation. If you just had potassium currents, it wouldn't work. Now, would it? All right. I guess I already talked about this, but this is a good sort of graphic it partially addresses her question. Accommodation and refractory periods. This gives you, again, some idea about the absolute refractory period. And actually, her point is correct in that the absolute refractory period, the way I've been talking about it, is really measured from the initiation of the action potential. So it really is sort of, it coincides with parts of the action potential rather than post-seeding it significantly. And there's some. So the relative refractory period is really the one that exists for a few milliseconds after the action potential. The absolute is really, you're right, it's closer to overlapping with much of the action potential. Good point, thank you. Now, there's some interesting aspects of accommodation and refractory periods having to do with the kinetics of sodium channel inactivation. And it gets to an important question about the kind of depolarization that's going to be most effective for generating an action potential. Turns out that if you want to get an action potential, if you want to get to threshold, the more rapidly you depolarize, the better. If you depolarize very slowly, you're actually probably not going to cause an action potential. This is a process called accommodation. The reason for that is because as you depolarize very slowly, many of your sodium channels actually end up in the inactive state. Okay, there's competing processes when you depolarize. Opening versus inactivation. When you depolarize rapidly, basically activation dominates over inactivation. When you depolarize at a very slow ramp, it actually turns out inactivation will dominate over activation based on their kinetics. And of course, potassium channels will also open with slow kinetics, and that will also make sure that thresholds can't be reached. So a very rapid depolarization is much more efficient for generating an action potential than a very slow one. And again, it has to do with the kinetics of activation and inactivation of the sodium channels. So they're very important. Okay. So I started teaching this class. I didn't actually know this one, but it's actually kind of interesting. I just want to talk about it for a second. This is an example of a membrane potential recorded from a single-celled organism, paramecium. You've all seen them. Very common little creatures in fresh water, sculling around with their little cilia. Turns out that you can generate an action potential across a paramecium cell membrane, very similar to a vertebrate one in that it's primarily dominated by sodium. It's actually a lot longer than a typical action potential that we're talking about. And it turns out they actually have a purpose. They regulate the beating of cilia. helping them avoid noxious cues. If you block the action potential, the main effect is the paramecium can't swim backwards. It can still swim forwards, and it can still engulf food. So this is just interesting for thinking about action potentials and membrane potentials in a sort of a more evolutionary context. Paramecia are a very ancient eukaryote, and they've been around a lot longer than nervous systems. So who knows? It's clear that these kinds of complex changes in membrane potential have been important for signaling for a long time in thinking about evolutionary time, and have evolved in things like vertebrates for different sorts of signaling, perhaps more complex types of signaling. But sort of the basic molecular machinery predates the nervous system, and the basic idea that you use it to signal important stuff also predates the nervous system. So it's just interesting to think about that in sort of an evolutionary context. I thought that was kind of cool myself. I didn't know that. So you can find that in action potential in a paramecium. So the next paramecium you find, try and record an action potential. You got to pin them down, though, so they don't move. All righty, it's noon, so we're done for today. We got through a bit, and we'll get through a bit more over the next couple of days. So hopefully you have a better understanding, if you didn't already, about membrane potential and some of the changes in membrane potential that are important for action potentials. All right, good. Have a good rest of the day. If they have announcements, they should announce them now.
Remember the meeting place, so it's back here in the deliverable. Also wanted to remind you if you have any questions, concerns, any directions again, you're always supposed to first use Dr. Millar and myself as your resources. Kathy also has places on Blackboard, so you can like digital copies and set up tomorrow. Any questions? Just a quick reminder. Thanks. It's Dr. Depper's birthday today, so we Brandy's on one of ours here.
I don't think I've met you yet. I'm Megan Dupper. Nice to meet you.
What's your name?
Megan Dupper.
Oh, hi. Nice to meet you.
Thanks for letting us.
I was done.
Thanks.
You're welcome. All right, if I sign, can I get a brownie?
You ate them. You should have chicken lemon bars too.
So I was kind of like raised to believe that the terminal neutron like sucked up neurotransmitters. Turns out like at the distal area is not up here.
It turns out that even pretty much anywhere you look, most of the uptake is in the brain. So the end feed of astrocytes in your brain, these end feed of astrocytes pretty much surround most synaptic specializations. And that's where most of the uptake happens. There is uptake into neurotransmitters, and there's also recycling, correct?
Oh my God.
So that is correct. But really the bulk of it is glial dependent.
Okay.
So you weren't wrong. You're missing a component.
Is it more like I could think of it? You said like more glials up here, but the reuptake for axons in the neuromuscular junction.
Well, remember, neuromuscular junction is enzyme dependent. So calling it cleave, then the reuptake occurs. So the actual inactivation of the neurotransmitter is the enzymatic cleavage reaction. Then acetylcholine are taken up independently, and that too is actually significantly glial dependent.
So there's
But it's different glia. It's different glia.
down
Oh
here.
yeah, like Schwann cells mostly.
Oh, I didn't know.
Yeah, you got plenty.
I thought they all.
No, you got glia all over the place. Schwann cells in particular are peripheral.
Okay.
Yeah, they serve similar roles in the periphery in many places.
Penumbra? What's that? What do you? What's
That's
the penumbra?
so.
You said going into the penumbra.
Oh yeah, sorry. So when you have a stroke, the primary site is where you have the infarct. You have a